Welcome, everyone. Thank you so much for joining us today. This workshop on the evolution of multicellularity is Janelia's 9th and final workshop in our 4D cellular physiology series. It has been organized by Jennifer Lippincott-Schwartz of Janelia, Zev Gartner and Wallace Marshall from UCSF, and Manu Prakash from Stanford. Each session will have a series of 10-minute talks with 5 minutes for audience questions after each talk. We really want to hear from you. So please write your questions at any time in the Q&A box, and the moderator will ask them during the Q&A periods. You are also welcome to use the chat box at the bottom of your screen to share your thoughts, comments, your big ideas. But please, if you have specific questions for the speakers, enter those in the Q&A box because that allows us to keep track of them. Each session will conclude with a 30-minute forward-looking discussion among all of the panelists in that session. As a reminder, recordings will be posted to Janelia's YouTube channel within a few days after the close of the workshop. And with that, I will turn it over to one of our organizers, Zev Gartner. Zev.
Thanks, Janine, and good morning, everyone. On behalf of Jennifer, Wallace, and Manu, and the rest of the organizers, it's a pleasure to welcome you to this symposium. Our goal in putting this together was really to bring together a number of eminent scientists and engaging speakers into a common place to begin a discussion on the general topic of multicellularity, how it's regulated and how it arose through evolution. We've outlined a number of questions that we might all think about throughout these presentations. These include, what are general principles of multicellularity? How can theory help us to identify these principles? What new tools do we need to test these theories? How do we think about aging and development and how they differ between unicellular organisms and multicellular organisms? And how can what we learn through answering these questions help advance applications, whether in biomedical sciences or in other areas of engineering? But more broadly, sort of beyond focusing on these specific questions, we want to begin to reveal how the questions that we're asking today may help inform upon the questions that we should be asking tomorrow. And so hopefully we can make progress towards that broader goal throughout the presentations. So again, thanks very much for joining us. And with that, I'll hand it over to Jennifer.
Thanks, Zev. Yes. I'm super excited to be part of this workshop. The origin of multicellularity is really, I think, a very, very exciting field. We still know very little about it. Our first session is going to touch on, I think, some very fundamental principles about multicellular organization, including the role of epithelial, the characteristics of epithelial cells, which seem to be a very core function of any type of multicellular organism. We're going to be looking at the role of forces in the behavior of epithelial layers. And we're also going to be taking a look at some of the most primitive multicellular organisms in that context. This is all within the first session. It's comprised of, we have 6 speakers, including 2 selected speakers from abstracts. I want to just dive in with introducing our first speaker in this group, Inaka Ruiz Trillo from the Institute de Biological Evolution. So Inaka, please.
Okay, can you hear me?
Yep, looks good.
Perfect. Okay, so, well, good afternoon or good morning, everybody. Thanks a lot to the organizers for inviting me to this conference. It's really a pleasure to be here. And what I'm going to do today is I'm going to provide an overview of how I see the future of the field of animal origins. This field, the aim of this field, is no other than to understand how organisms, unicellular organisms such as these ones, could eventually give rise to the huge diversity of animal body plans. We are in the early innings of this field, but what remains ahead is, I would say, huge, huge and profound. But I do believe that if we address 3 different types of questions that I'm going to mention now, we will be able to bring this field where it belongs to be. These 3 types of questions are, first, the questions that we all want to address. These are natural questions. Second, the questions that we all want to address, but we are afraid to ask. And the third one, my favorite, are the invisible questions, the unseen questions, those in which we don't even know there is a question. So let's start with the first one, the questions that we all want to address. And in order to see that, we need first to see where we are in the field and where do we want to go. Where do we want to go is here. It's in the how. It's in the how unicellular ancestor became the first animal. That's where we want to go. But in order to go there, we first need to unravel 2 different ancestors, the unicellular ancestor and the first animals. And to unravel the unicellular ancestor, this is very important, to unravel that, we can only do it if we compare animals with the closest unicellular relative. And the thing is that, and that's why this is an early field, because we didn't even know 15 years ago who are the closest unicellular relative of animals. So the first stage of this field was to unravel that. We didn't have a good phylogenetic framework about the origin of animals, but now we have, now we do. And we know that there are 4 unicellular lineages that are closely related to animals. And we also know that these 4 unicellular lineages are quite different one to each other. Moreover, all those 4 unicellular lineages, they all have temporal multicellular stages, such as, for example, the colonies of choanoflagellates, or the multinucleate cenocytes of ichthyosporans, or the aggregates formed by cell aggregation of Capsospora and other phylasterians. One of the questions that was posed on this conference is the difference between unicellular and multicellular organisms. And there are many. But I would say that the most important difference by far of unicellular versus multicellular organisms is the knowledge that we have on them. We know much more about multicellular organisms than we know about unicellular ones. And this creates a problem, and this creates a bias that affects our understanding, our approaches, and our interpretations. So the first thing we need to do is to break this asymmetry that has been here for far too long. And we have actually done that for something, for the genomic data. And we did it, we broke this asymmetry because we had a very clear objective at that point. We wanted to unravel the genomic composition of the unicellular ancestor of animals. And we knew we could only do it if we had genomes of unicellular relative of animals. So between the lab of Nicole King and our lab, we have been able to obtain 13 genome sequences of unicellular relative of animals, comprising all these 4 different lineages. And what this data is telling us, what those genomes are telling us, is that the unicellular ancestor of animals was genetically quite complex, much more complex than previously thought. Yes, we see an innovation, a continuous innovation of genes from the ancestor, the larger ancestors, to the origin of animals. And yes, we see a little bit of significant innovation at the origin of animals, but we also detect that many of the genes and pathways that were thought to be specific to animals, and this is what we thought only 14 years ago, now they are not specific anymore. Now, this is the view. So a lot of the genes, families, and pathways that we thought specific to animals are present in the unicellular ancestor, that were present in the unicellular ancestor. This is telling us that the unicellular ancestor was genetically quite complex, but also suggests, very importantly, that co-option played an important role in animal origins. And to understand co-option, to understand how these genes were recycled for new functions, is one of the questions that we all want to address, and it's one of the questions that I think we should be addressing in the future, and it's also one of the questions that we can address now and we couldn't in the past. And why can we address now this? Because we are developing genetic tools. And importantly, we are not developing genetic tools in just one taxa, because that will be wrong. We know, and we know very well, by analyzing genome data, that only one of those taxa, comparing only one of those taxa with animals, we can get inaccurate results. That happened actually when we were comparing the genomes of animals with the first genomes of guanoflagellates and reconstructing. I'm going to just show two examples, the origin of integrins. We thought the integrins were specific to animals because they were not present in the first two genomes of guanoflagellates. But if we have more genomes, the story is completely different. And integrins were secondary loss in fungi and guanoflagellates. And the same with several transcription factors. They were secondary loss. So we know this is a good example that we need broad taxon sampling. And we have it now. Thanks to the efforts of several labs, we now have genetic tools in several of these unicellular relatives of animals. So now we can address the questions that we all want to address. Let's go to the questions that we all want to address but are afraid to us. And I think the most important question here, there are several. I think the most important one is to go beyond the origin of animals and try to understand, to have a common ground, a common understanding of the origin of multicellularity. So what do they have in common, all these independent acquisitions of multicellularity in the tree of eukaryotes? Do they have something in common? Are there mechanisms in common or not? So we need to do that too. And this is what we need here. It's actually to bring together fields that classically have not been speaking one to each other. So we need to bring them so that they can engage together and we create a common ground. And then we go to the third type of questions, the invisible questions. And I'm going to use an analogy here. So if we consider the origin, the creation of a field, like building a house, first type questions is going to the details. So, for example, you can define very well how is going to be the gym of my house or the swimming pool or the kitchen. This is cool research and fancy and visual and very publishable. So it's very good. But then you also need to figure out what are you going to do with your house, with your field? Are you going to are you going to communicate with other fields? Are you going to create better neighborhoods? Or are you just going to use your swimming pool for you and that's it? And that's where we need to go, for example, in order to to go beyond the origin of animals and go to the origin of multicellularity. But then we have the third type of questions, and this is the basis. This is going to the basis. This is seeing what we couldn't see before. So imagine we are delineating, we are trying to find how are we going to have the swimming pool or the gym or the kitchen. But nobody has asked whether we can create a house in this spot that we have prepared here. We are assuming we can because it's a neighborhood full of houses. We are assuming we can because somebody asked us, but maybe we didn't go to the city council to ask. So we need to go and ask, because if we can do it, if we cannot build it here, we have to build it in another place. Usually these problems come by assumptions, by assuming things. And here, we may be assuming that what we see is what it is. We may be assuming that what we know is what it is. And sometimes what we know and what we see is what it is, but not always. And I think we have a lot to think about this. We know this is what we know now. But come on, nobody has systematically asked whether there are lineages that are in between guanoflagellates and animals. Nobody has systematically searched for them. Actually, most of the taxa that we know here were found by luck, serendipitously. So nobody has searched for them. And we also know by environmental data. Environmental data is suggesting us that there are some of those lineages here in these positions. So I think we need to look for them because, as I have explained before, having different taxon sampling, having different taxa can completely, dramatically change how we interpret the unicellular ancestor or the first animals. And if it changed the reconstruction of the ancestors, then it changed the how. Then it changed how the unicellular ancestor became the first animals. And I think we need to go into that. So I believe that if we manage to address all these three types of questions, we will be fine. We will create a long-lasting field for people to work and grow and enjoy. I also think, that's true, I also think that if we fail in some of the questions, we will be fine too. But then the future generations will try to address that. And you know what? I rather prefer that we do it now and we do it right. Thanks a lot. I'm glad to take any questions.
Great. Thank you so much, Inaka. I'm going to start off the questions while people are writing down their particular ideas, et cetera, with the question of, and this is maybe sort of an invisible question on in your concept, and that is, do you have, from the type of analysis that you're doing, any understanding of the types of organisms that the particular organism that you're analyzing in this genetic tree, how they're interacting with each other? Because we know that animals are evolving in a complex world environment where there are other animals, there's plants, there's bacteria, there's archaea. Can you get a sense of sort of an interaction sphere that any of these particular species are interacting with from the data sets that you're gleaning? And do you see this as relevant?
Yeah, thanks. So we know by the work of the lab of Nicole King that there is important interactions between quanoflagellates and bacteria, for example.
Yeah.
We don't know much of the other organisms, but the problem is we haven't studied that. You know, I'm sure there is interaction. So actually, if you think about a unicellular organism, they have to interact in many, many different conditions. Actually, much more different conditions than a cell inside a multicellular organism. So they have to deal with.
Can the genetic analysis, can your genome or transcriptomic analysis in any way give you clues as to what that interaction sphere is?
I don't think so. I think the only way to get it is by, comparing metabarcoding information with other metabarcoding and trying to see whether there are organisms that interact at the same time and at the same space, or going to the, natural history and the biology and trying to understand much better these organisms, the biology of these organisms, which is highly needed.
So I'm going to go quickly to these questions. One is by Rensky Varumans. Thanks for the interesting talk. How would we go about studying gene co-option? What would be an interesting question to ask there?
I think we have a lot of interesting questions before we go to a conceptual framework of co-option. So we need to understand the co-option of specific genes in order to understand how they were recycled. And there are many genes that will be very interested, like genes involved in cell addition, genes involved in cell signaling, and of course, genes involved in cell differentiation, and also different signaling pathways. So if we unravel those, we can have a hint of what was the potential function and how was that recycled. And then later on, once we have some of that, then we can have a more wider conceptual framework of how co-option play a role in the origin of animals.
And next question is Olivia Barbosa. When you see signaling pathways with missing links, what do you think about this? Are they non-active signaling paths? Are there genes with unknown functions? Or are the signaling pathways more simple in these groups?
I actually think everything that she mentioned as possibilities. I think they are all those potential, it can happen, all the things that she said. And we just need to do experiments and then see what's the answer. Let us, the organisms inform us.
And David Booth's question is, in some of your environmental sampling, you have uncovered some unexpected diversities in
quanoflagellates. Yeah.
Do you believe more sequence information may place them outside of this species and sister to animals?
I think there is the potential. So one of the problems that we have with metabarcoding is that we are analyzing a very short section of the 18S ribosomal DNA. And then the phylogenetic information is not very good. So it just tells us that there is more diversity. That's what I think we need to do, a systematic approach to this question, and with wider techniques. and see if we get something. But I believe there may be important new lineages there, and nobody has searched for them.
And our last question is from Manu Prakash. Manu, do you want to unmute?
Yeah, Inaki, thanks for such an incredible talk. My question is piggybacking a little bit on what JLS was referring early on, but thinking about the environment a little more deeply. What do we really mean when we think about understanding an organism? And most often, many of our techniques bring these organisms outside of the actual environment into our labs. And I'm just curious, just from your perspective, and as we expand studying more number of these unicellular organisms, we rely more and more on genomic data, but we forget the strange environments that they have evolved in. And really, to understand them, how do we really bring the science to the environment rather than the other way around? So I'm just curious on, as you embark and many of us embark on searches for these new animals, how do we do justice to the study and don't repeat the mistakes we have made for many common model systems?
Yeah, I think that's a blind spot that we have. It's a huge blind spot. You can have a big track and you don't see it, but it's a blind spot that is very difficult to get off. And the only way that you can get off, I think, is to get more and more understanding, even if it's in the lab, because in the lab, you can change a lot of the conditions. So you need time, you need people, you need efforts, you need labs to go farther and farther. And of course, we can try to do more in the biology, in the environment, but also in the environment, if you spot an environment, it's one specific moment, one specific organism. So we don't know the whole life history. And I think most of the things that we don't know is because we haven't seen it. It's not because they are not there. So, I didn't have time to explain more, but we are discovering, we and other labs, we are discovering a lot of fascinating features and morphogenetic movements and things about these organisms. And this comes just by observation on the lab. And the more we change and the more we analyze, the more we will try to put this blind spot there. But we will always have this blind spot, I'm afraid. The important thing, sorry, I want to finish because I think the important thing is to be aware that you have this blind spot. One of the worst things that can happen in science is that you have a blind spot and you are not aware of this blind spot. If you are aware, then, that's fine.
Well, thank you so much, Inaki. We've got to move to our next speaker. There are many questions in the Q&A panel, so please go through them. We can bring some of those questions up again at the conclusion of this session when we have our big discussion. So I want to next introduce Keiko Tori from UT Austin.
All right, thank you so much. This is exciting. Can everybody hear me?
Yes.
So my group is interested in studying functional tissue patterning in plants at the interface of cell fate specification, cell-cell interaction, and cell polarity in plants. So plants does not have any cell migration. So everything is communication and division control and the cell shape and the polarity, etc. Especially we are looking at the leaf epidermal development, where as the leaf grows, the surface single cell layer generate two cell types. They have de novo stem cell popping up to generate this stoma or stomata that can open and close to adjust the gas exchange while minimizing water loss for plants to not wilt. At the same time, this stem cell also produce pavement cell, this jigsaw puzzled, tightly intercalated cells that protect plant's body from desiccation, heat, stress, pathogen entry, all sorts of environmental stress. And essentially, this is a fate cell decision problem that the initial cell do asymmetric cell division and generate two daughter cells, and the only one of which can become stomata, and the other remaining will become pavement cell. And the density and the distribution of stomata is critical for plants for maximum fitness in a given environment, as plants cannot move, so they have to cope with different environment. They do adjust the number of stomata as they develop new leaves. And through the decades of study of my group and others, we know the cell state transitions and how master regulators regulate each fate and how cell-cell signaling enforce which cell to become a winner cell, etc. But there are so many questions still unanswered. For example, during the stomatal stem cell divisions, there's a cell polarity switch that occurs to allow this kind of a configuration of two stomata. But even though many scientists, such as Joan Don's group, has elucidated the polarity component that is unique to plants, because plant cells does not divide like animal cells, the mechanism of this polarity switch is still unknown, for instance. And also, this is the wrong time, but as the two daughter cells happens, if you ablate the winner cells, the loser cells, the neighboring cell could take over stomatal fate. And this is mediated by cell-cell communication, but the exact understanding, see, we kill this one and the remaining one become winner. But this kind of interaction is still unclear how that happens in correct time and space. So I have been obsessed with visualizing the decision-making process in time and space in this process by mostly exploiting the peptide system that the two antagonistic peptide that make stomata or inhibit stomatal development by acting on the same receptor. So we try to see that how the receptor internalizes or what the subcellular dynamics after addition of different antagonistic peptide that gives two contrasting effects. Or we even try to tie up with the physicist, astrophysicists, to use their autocorrelation program, which is a program to map the galaxy in the space. We utilize it into the millimeters of tiny Arabidopsis leaf epidermal cells to generate the genetic mosaic of sectors that produce one of those antagonistic peptide and see how the two-dimensional pattern of stomata deviates along the inside as well as nearby sector to see what are the effective diffusion range of these peptide. And actually it matches with the Turing patterning. But you can't really see how peptide actually diffuse. So we try to collaborate and develop FLET sensors to see which cells are receiving the peptide by activating the downstream cytoplasmic MAP kinase cascade. Or we even collaborate with a synthetic chemist to generate peptide that has individual multicolored. So you can see apply like a bunch of different peptides with different color and see how cell reach to the decision-making process. But still, this is all artificial because that's not the natural peptide that is occurring and the plants are producing either. So thinking of what are the big questions in the plant developmental biology field for the multicellularity, and this is my very biased view, so I just want to emphasize that this is my point of view, but I think that one of the key questions that we are still trying to understand and not really understanding is how cell fate is coupled to the intrinsic cell cycle, cell division, as well as that of the neighbors, like cell division or all sorts of activity of neighbors, including physical or mechanical contacts. And group like Minako Ueda's lab in Tohoku University, Japan has been doing amazing, I think I need to put it back, amazing live imaging to see how embryo patterning occurs throughout and how the cell fate is acquired during this stereotypical series of asymmetric division that happens in the absence of cell migration. And these monitoring really give us the hint, as well as using the drug screen to disrupt a particular process to understand what is determining the key in the fate determination process. And coming back to my own system of this, whether you want to become stoma or pavement cell, we find that experimental manipulation of the cell cycle slow down. So if we extend a particular type of the cell cycle called the G1 phase, then we found that actually stomata become kind of like a hybrid cell between pavement cell and the stoma. So how could this cell cycle really regulate the specific fate of stomata or pavement cell? That's also kind of unanswered question. And we need a lot of things. Maybe it sounds generic, but we need better live imaging tool to push the boundary, and optogenetic, optochemical, or synthetic tool to manipulate such dynamics process. And I'm not a genome person, but integrated 4D single cell analysis or type of the map is critical, I believe. And the most largest question in the plant biology field is what makes up the plant as a multicellular organism, and what makes a plant an individual organism? This sounds like a very cumbersome question, but the plants are very strange in a way that every cell essentially has a potential to capacity to differentiate. You know how easy to propagate plants. You can just grind the cell in a juicer or mixer to make a green juice, and the individual mesophyll cell can become individual plant. As well as if you just cut it off the root, that can reinitiate quickly the stem cell niche and just grow. This is because of the lifestyle of plants. They can't move. You can come and grab them, torture them, but they have to survive. So they have this enormous capacity for regeneration, as well as their enormous flexibility for organ fusion or grafting. You can actually graft the arms or legs of the different species or genus, order, or even family. So here is the example that Arabidopsis grafted with tobacco and the tomato, and they are actually tomatoes growing and making tomato fruit. So what are the individual boundary of species or genus or order or family? And this can be even done within the phyla, that you can connect moss and tobacco, for instance. That is from Notaguchi's work. The people finally start understanding single cell or the gene expression or transcriptomic analysis to understand what's going on and what makes them plants so flexible in terms of accepting the others to generate multicellular organisms that is functioning as an independent system. For the same line, plants live so long. Some plants live like thousands of years, and what do they think they are the independent plant, or they are already the part of the different plants that behaving as independent organisms? We don't know, and that's a big question, because plant does not have any centralized system like our brain, but their growth is yet highly coordinated. So in this case, this is a beautiful, if you wound, then plants can signal right away. These are calcium and others, but plants knows that they have to coordinate and behave. But at the same time, I still wonder, what is the independent plant when you think about plants? Because if you think about strawberry, I'm not sure how many of you grew strawberry in like a pea patch, but they actually make runners, and each runner can make strawberry, kind of like a strawberry shoot, and they are connected. Clearly, this is a single organism, right? Single, like me. But if you cut it out these, then they become independent. I suppose they become independent shoot, and that's how you propagate. But there are many hormones, small RNA, electric signals, so many things are going back and forth. So what makes them an independent plant or individual organism, or part of the organism, supra-organism, is one of the big questions that I'm quite interested in, and the plants can address something exciting. So to sum up, so understanding multicellularity through the lens of a plant, that how cell fate is coupled to the intrinsic cell cycle, cell division, as well as that of the neighbors, and most importantly, what makes up a plant as a multicellular organism and what makes it an individual organism. So really, to understand this unique and successful strategy of plant as a multicellular organism will provide, I hope, to provide the broader view on the origin of multicellularity. And we need more collaboration opportunity with physicists, chemists, computer scientists, engineers, and invisible question, as mentioned by Iñaki, but I think I need to learn more from classic botanists and zoologists, because there are so many knowledge that ancient, I wouldn't say ancient, botanists and zoologists found that I don't, as a trained molecular geneticist, I don't really appreciate. Okay, that's it, and thank you. And I accept, I'm happy to have any questions. Thank you so much.
Thank you, Kiko. That was really very exciting talk, very, very interesting. We got some questions coming in already. First question from BuzzBomb. Are there plants that have very limited regeneration and that have a germline that is set aside early in development? Also, are there plants that have a relatively fixed size and shape?
So fantastic question. And thank you for asking, because I realized I rushed through the 10 minutes talk and I forgot to tell you one super important thing, that the plants do not have set aside germline. So plants didn't maintain as an individual organism, live thousands of years without having cell migration, but also without setting aside the germline. So that's one of the most fascinating questions So for instance, so I had that in the original talk and I removed to save the time, but great that you asked the question. So recently there was a paper published because of the advanced genomics. You can actually genome sequence the individual branch of these kind of majestic trees that lives hundreds of years, hundreds of years, right? So there was a paper published from a Switzerland group that they used the Napoleon oak, which is an oak growing in a campus of the University of Lausanne or something like that. And that's because history knows exactly the age of that plant. That's like 234 years old, I think. That's the time Napoleon visited that place or something like that. Anyway, so they sequenced the individual tip of the leaf coming from individual tip of the branch and see how much somatic mutation exists by like whole genome sequencing. And they actually find surprisingly little mutations where the SNPs are found among the different tip of the different branch of this ages of old big oak tree. So the idea is that the plants had something called meristem or the shoot tip that keep making leaves or branch or flowers. So some of my biology colleagues said, is there anything interesting in plant development? You just water plants. So, but it's amazing that the things keep coming up and you don't even know where those leaves comes from, but they are from the stem cell at the tip that keep divide slowly as long as the plant grows for many of hundreds of years. So this sequencing study suggests that how those stem cell niche are protected from some type of somatic mutation or genotoxic stress, maybe physically or some molecularly by some sort of stronger mechanism for DNA repair and as such. So that's one of the interesting things that we can learn certainly about the longevity and the protecting the somatic cell as plants grow such a long life as a multicellular organism. And sorry, there was
another, I'm just going to move to another question. This is from Merlin Staps. Thanks for an interesting talk. To what extent do plants' flexibility manifest itself at the cellular level? Are cell fate and cell differentiation reversible?
Oh, so that's an interesting question. So plants, unlike animals, kind of have to work hard to stay differentiated. So they are very easy to revert. And latest studies, like this is not my work and maybe unpublished, but people try to monitor all the epigenetic state to see what correlate with the reversion, fate reversion, or the initial reprogramming. And it appears that the histone acetylation is playing a key role. And it seems like that the universal in the many different system of plants during regeneration or the fate reversion from terminally differentiated stomata into the initial epidermal cells, protodermal cells. So that aspect might be similar with animal as well, that how epigenetic state is critical for maintaining the cell fate, but how easily that can revert or stay terminal, etc., might be quite distinct between plants and animals.
I just want to throw out a question myself related to your work and just this bigger concept of interplay between plants, animals, bacteria, archaea. I mean, plants are eaten by animals. Plants create defenses against animals. Animals basically can grow, can play, not just humans, but lots of animals control plants in terms of how they're growing, et cetera. What role do you see that playing in evolution? This interplay between animals that are fundamentally reliant on plants for survival or plant products at some level in the evolution and the processes that you're describing for these plants?
Oh, that's a great question. That's a very big question. And as a like a plant biology, intro biology lecturer, I often favor to say that the plants really don't need animals, but we absolutely rely on plants. They are the one who came first to conquer the niche in a land around 450 million years ago, providing the niche for animal to evolve as well. So that aspect, I feel like it's it's like we have one-way love to plants because we really need them, so we have to be nice to them. Then that's why we all like green and forest. But that's my philosophical view. And in reality, I think it's such an intricate relationship that having both, or including microbiota, which is becoming very in, like a gut microbe-like thing in the soil, the microbiota actually establish plants condition for them to thrive as well. So those are really shaping all together. And the plants are sometimes like a supracellular organism, like a bamboo root, they make a huge network of acres of land and behave as a single organism. They flower at the same time and stuff like that. So I think they are super interested in, and of course, they are probably fundamental for the co-evolution of the plants and the animals and the other microbes.
Yeah, wonderful. There's some more questions in the Q&A. We'll pick them back up when we come into the discussion, but we need to move on in the interest of time. Our next speaker is Lucy O'Brien from Stanford University. Lucy, do you want to hook up?
Excellent. So thank you so much for this opportunity to speak. And I want to start by thanking this awesome team of folks here whose work I'll be sharing with you today, particularly Irina Kolotueva, our collaborator at University of Lausanne. Irina is responsible for the incredible EMs that I get to share with you. So if we're talking about multicellularity, then in my view, and as Jennifer mentioned at the outset, we have to talk about epithelia. Epithelia are the most ancient type of multicellular tissue, and the whole raison d'etre of an epithelium is to create a barrier that separates an inside body cavity from the outside world. Now, this was beautifully introduced by Inaki. Some of the earliest forms of multicellularity, like colonial sponges, these are single cell organisms that join together to form a sort of proto-epithelium, which creates a cavity that acts as an external gut. Hydra and other early metazoans, they went one step further. They created a permanent gut epithelium with a single opening to the outside world. And later metazoans really exploited an epithelial barrier function. They formed sealed compartments and tubes that evolved into highly specialized and complex organs. To me, this image really captures how amazing epithelial barriers are. What we're looking at is a cross-section taken through the adult Drosophila gut. On one side, there's bacteria, gastric acid, ingested food, all the things in the gut lumen. And on the other side is the interior body cavity of the fly. Between them, there is a single layer of epithelial cells that's charged to make sure that these two sides never mix. Epithelium can fill this tall order because they have two special features. They have a protective apical surface that's shown here in pink. It's folded into microvilli, covered in a glycocalyx, studded with immunopeptides. This apical surface is the first line of resistance to the outside world. The second feature is occluding junctions. They're shown here in blue, and they're at the lateral edges of each cell's apical surface. These junctions stick cells together, forming a leak-proof seal between them that prevents anything from passing through. And these are known as tight junctions in vertebrates and smooth septate junctions in the fly gut. So epithelial tissue architecture is pretty amazing. But what's even more amazing to me is that these epithelial barriers aren't static. Throughout our lifetimes, many of our epithelia continually self-renew. They're replacing their constituent cells over and over again. But how do they do this? And particularly, how can they add new cells without jeopardizing this vital barrier? Well, a starting point is to consider where exactly these new cells are coming from. In many barrier epithelia, they're coming from progenitor cells, like these little yellow cells here in the fly gut. These progenitors aren't fully fledged epithelial cells. They're basal to the barrier. They don't have an apical surface, and they don't have any contact with the outside world. And so how does the cell go from this to this? The question isn't relevant just for the fly gut. Many adult barrier epithelia have basal progenitors and thus have the same architectural challenge. And this situation comes up not just in adult organs, but also in embryonic development. And it turns out that for one of these embryonic tissues, the frog skin, we know how this occurs. And this is beautiful work from other labs showing that it occurs through a process called radial intercalation. Here, a progenitor cell moves basal to apical as it differentiates. And as it moves apically, it contacts this apical network of occluding junctions and forms a new junction with it at its tip. This junctional contact then targets the new cell's apical surface, which, if you were watching it from above, this apical surface starts as a small dot and then grows radially outward. So radial intercalation is how one epithelium, an embryonic epithelium, does this. And now we can ask whether an adult epithelium, the fly gut, does this as well. We use correlative light electron microscopy to identify early stage progenitor cells using a fluorescent marker of terminal commitment. We then looked at what was happening with these cells occluding junctions. Our progenitors are born without junctions, but when they commit to differentiate, we find that they make new junctional contacts with their mature neighbors. So here, the terminal progenitor is red, and its new junctional contacts are these 3 green points that are at the tips of the fingers here. Each green point connects with one of the pink or purple junctions that belongs to each of the progenitor's 3 mature neighbor cells. So progenitor integration in the fly gut starts off looking like radial intercalation. But when we then looked at older progenitor cells, we found that this is no longer the case. It's in older progenitors that the apical surface first appears. It's here in blue. And remember that in radial intercalation, the apical surface starts out as a dot and it grows out radially from that dot. Well, here, the new apical surface forms this concave structure. And intriguingly, the structure seems to be sitting underneath a large occluding junction. Looking at 3D reconstructions, it's clear that this concave apical surface in the new cell, this is completely distinct from the lumen contacting apical surface of the epithelium. You can see that there's a clear gap right here. But what is this surface really? Well, to understand this, we turn to Irina. And what her EM tomography revealed is that these structures are deep, microvilli lined invaginations of the new cell's plasma membrane. Here, that progenitor cell membrane is in red, and this cell has 2 of these invaginations in blue and pink. We call them pre-assembled apical compartments, or packs. Here you can see how these packs are covered by this green junctional blanket that the progenitor cell has made with its overlying mature neighbors. There seems to be an opening in each of these packs. It's not an opening. What that is where the pack is covered by the plasma membrane of the overlying mature cells that are not shown here. So these packs are actually intercellular lumens. So what we have in the fly gut is not radial intercalation, but a distinct mechanism. The new cell's apical surface, this pack, forms in the shelter of a junctional niche. And when the pack is sufficiently mature, then this junctional niche retracts, and the pack becomes part of the gut's luminal surface, which integrates the new cell with the rest of the tissue. So 2 mechanisms toward the same end, radial intercalation and PAC-mediated integration. I'll suggest that these 2 mechanisms lie on 2 ends of a spectrum. Radial intercalation is fast. It happens in minutes or hours, but the progenitor needs to expose its new apical surface immediately. Whoops. Now, PAC-mediated integration, on the other hand, is slow. This happens over 2 to 4 days. But here, the progenitor doesn't expose its apical surface until it's ready for the outside world. Perhaps the choice between these 2 options depends on physiological context, whether you're protected inside an egg, like an embryo, or exposed to a harsh external environment, like the adult gut. And to close, I'll offer that perhaps it's not just form, but also formation that follows function. We're all familiar with convergent evolution, where species evolve similar structures through different paths. Perhaps we can also think about convergent morphogenesis and speculate about what equivalent sorts of evolutionary forces might be acting on cells here. And lastly, what can Janelia do? Well, given the hundreds of hours that it takes to trace EM tomograms by hand, I would love to see development of algorithms to recognize junctional structures between cells and trace them automatically. I showed some CLEM, but how about combining spatial transcriptomics with EM? This could cast new light on longstanding chicken and egg questions about cell fate and cell morphology. And finally, I haven't shown live imaging, but improving image registration algorithms so that movies of complex tissue scale movements like peristalsis, these movements can be visualized at sharp cellular resolution. I think this would really open up a truly new morphological realm for understanding physiologically active tissues. Thank you, and I'm happy to take questions.
Thank you so much, Lucy. That was really exciting. We got some questions for you. The first one is from Wallace Marshall, who's asking, is the PAC entirely intracellular? Is membrane fusion required when it becomes the actual apical surface?
Yeah, Wallace, that is really fascinating because we spent a year thinking that these PACs were actually vacuoles inside the new cell. They are not vacuoles inside the new cell. They are a highly asymmetric intercellular lumen in which the partner cells that make up the lumens have radically different contributions. So 95% of the surface area of a 2-cell lumen comes from the new cell, and it's deep membrane invagination that forms that kind of koosh ball inside that looks like it's inside the cell. And then the mature cell, it's a partner in this lumen, but it hardly does anything. It's just its membrane on the other side of the lumen just sits there like a blob with no supracellular structure.
Zev Gardner has a question. Have you looked to see what factors the neighboring mature cells might be positioning or depositing into the PAC? Does any cell-cell signaling occur in this compartment?
Yeah, we have wondered about that a lot. What we see in some of the EMs, including the one that I showed, but it went by at lightning speed, we see strands that look like strands of occluding junction that have been peeled off. Our hypothesis right now is that the junction forms first between the 2 cells, and we get these junctional septa forming, and that the PAC machinery is targeted to this site and delaminates the junction at the site that it forms. What else is in these? I don't know. We think that it's a closed compartment though. So yeah, would love to know more about that.
Lucy, just quick related to that. Have you looked at where the centriole is localized?
Oh, that's a good question. No, actually we haven't, but we obviously have.
It would be
the data.
very interesting to look to see if that centriole is right up near there.
I would love to talk to you more about that. Okay. Yeah.
So Eric Sidge's got a question. Eric, if you want to.
Yes, thank you. There seems to be a related phenomena that a group in Michigan examined, which when stem cells go from do EMT and then MET, a lot of the apical resident proteins are encapsulated in what they called an apicosome, which allows them to change from epithelial to mesenchymal and back. Is your structure similar, do you believe?
It is different, and that is right. And I'm blanking on the name of the group right now.
Deb Gamuccio.
Yes, Deb Gamuccio. Beautiful work. We thought we're dealing with that for a long time. Their apicosomes are they're intracellular vacuoles. Enrique Rodriguez Bulan and his group also recognized similar structures when NDCK cells in culture become depolarized. And this was back in the 80s. We thought we were dealing with that. And so it was mind-blowing to us to realize these are actually lumens forming between two cells, highly asymmetric lumens.
Thank you.
And just one quick last question from me again. Is this in any way analogous to what you think might be going on in plants? We've heard from Kiko in terms of the way plants, they create an epithelial, and they also, have stem cells that are sort of there looks like there's similarity in terms of. Do you want to comment on this along with Keiko? Yeah.
Yeah.
Keiko can chime in. Yeah.
Yeah, and Kiko, I loved your talk. What really struck me in looking at the stomata is their distinct membrane shape and curvature. And so in thinking about formation of these packs in our progenitor cells, it's clear that they have to commit to a terminal fate before they start to can assemble the machinery to make this deep invagination. And so this link between a cell fate decision and then a specific cell membrane morphology, I think how that plays out is really interesting to me, and wonder.
Yeah. It was super fascinating talk. I learned so much from those epithelia and the animal, more advanced the field actually. Plant cell actually also had a cell wall, which the intersection of the cell wall and the cytoskeleton and the microtubules govern the final shape. So once it's formed, it's not as flexible. So they have like a one-way path. But also, so that's also many unanswered questions, like a lot of people are interested in this jigsaw puzzle shape, and many, for instance, scientists studies that how could they form, and they actually, like two neighboring cells actually talk to each other with some sort of chemical signal and see which side become the lobe by making, putting more vesicles to the membrane and the increased membrane size as well as newly formed cell wall and stuff like that, while which side will become like a dip or whatever, the notch and the lobe and the dip. And they actually intricate communication is going on too. So when cell shape change, it's not just one cell, but also neighboring cell, right?
Yeah, actually, and that's another key point. Maybe we can talk more in the discussion. In both cases, the neighbors have to help, assist, accommodate.
Yeah, so we kind of automatically think if like a balloon, if one cell becomes big, the other automatically becomes small. But there's actually a calculated communication and deposition of the extra membrane material or cell wall components and many stuffs. So thinking more and more is just how biology system is unbelievable. I feel like more I learn, more I get like old.
So we need to move on now. Thank you. This is a really great discussion. We'll keep going later on with it. I want to now introduce Eduardo Marino. Please share your screen.
Hello, everyone. Thank you for the invitation. So my lab is focused in a phenomenon called cell competition that has implications for several bigger fields, including neuroscience development, aging, regeneration, and multicellularity. So we have seen the very nice talks of Iñaki, Keiko, and Lucy, talking about plants and animals. And it is known that many of these multicellular organisms actually evolve multicellularity at different time points and independently. So one could argue, okay, then what we learn from one of these groups is not going to be applicable for other groups. But one could also argue that there are common problems to solve, right? Every multicellular organism has to solve how to cooperate, how to make cells that were not cooperating before cooperate and write it in the DNA, and how to identify those that don't cooperate, maybe because they just cannot do it. I think this classical experiment with Pseudomonas tells us a little bit these problems that are common to long-term successful multicellular colonies. So Pseudomonas are a little bit in between unicellular bacteria and multicellular or transient multicellular bacteria. So when they have enough oxygen in the media, each bacteria is an individual. They don't cooperate. But when there is no oxygen in the liquid media, then bacteria start to cooperate to create this mat on the surface made of protoglycan that the cells are secreting, and then they can reach the atmospheric oxygen. But because they cannot tell who is doing it and who is not, start to appear mutants, defectors, that essentially do not contribute to the production of this protoglycan. At some point, these defectors also, because they have a competitive advantage because they are not using resources to produce this protoglycan, essentially the whole colony, the whole multicellular organism collapses and disappears. So I think this is very nice as an example of what are the challenges that multicellular animals have to solve in order to cooperate and maintain long-term cooperation. So that's the interest of my lab, how animal cells in particular found a way to cooperate. And in order to understand it, we decided to focus on what goes wrong. So what happens when cells stop cooperating and start competing with each other. So over the years, we and others have found three types of competition among cells. The first one is what one could call trophic competition for limiting amount of resources like nutrients, oxygen, or even more common competition for survival factors. This is very similar to a Darwinian competition, and I will not talk today about this. Then there is a second type of competition that is a cell selection mechanism based on marks, molecular marks at the surface of cells that allow cells to actually decide which cell remains in the tissue and which cell does not. So I will talk most of the time about the second time, but just to mention another type of competition that could also be relevant for multicellularity is how cells compete for space, what's called mechanical cell competition. And essentially in tissues where there is too many cells, or in tissues where there is compression among cells, to maintain the cell pool or an organ size and an organ shape, some of the cells are eliminated mechanically. So the story I'm going to tell you today was started by Krista Riner in my lab, a very talented Swiss postdoc, and then continued by Isha Madan and Rajan Gonya. And what they discovered in the lab is that just like we as humans can detect fitness decay in our colleagues by just looking at the graying of the hair or the wrinkles on their faces, cells also have a mechanism that is conserved from Drosophila to humans, where they can look at each other molecularly using a family of proteins called the flower proteins. So the flower proteins come in with two functions. Some flower proteins are going to tell the neighbors that they are fit and healthy and they have no problem. And some other type of flower proteins that we call the flower loose proteins will signal to the neighbor that there is a problem or they have damage or stress. But these proteins are very similar. Basically, they maintain the core domain and they only change the last bit of the protein. But this completely changes the message that they are telling each other. What has been also quite interesting is that there are many insults that a cell can have. For example, slower division rates or slower growth, effective polarity, incomplete cell clusters, even just cells that are totally fine, but happen to be in the wrong organ. So probably through different mechanisms, this will lead to this change in the flower isoforms they are showing, and they will start to produce the flower loose isoform to signal to the neighbors that that's something wrong. As I said, this is conserved from Drosophila to humans. In humans in particular, there are 4 of these flower proteins. And through experiments that I will not detail in much, but basically using CRISPR and then transfection of a single isoform, we could produce populations of human cells, epithelial cells, that are expressing either green or red fluorescent protein and are expressing a single of these isoforms. Interestingly, when you have homogeneous cultures of these cells expressing a single isoform, it doesn't matter if the cell is expressing the win or the loose isoform, they are all surviving and proliferating at the same rates. But when you do co-cultures of these populations, what happens is very different. You can see here we have one of these co-cultures. I'm not going to show you, of course, all the combinations, but in some combinations, what we can see is that now some cells that are expressing, in this case, the green cells that are expressing the loose isoform start to be recognized by the neighboring cells and eliminated. I will play the movie again. So you see the green and the red cells are interacting. At the beginning, they are fine, but then you see these cells start to die, the green cells. If you do the co-culture without letting these cells touch each other, nothing happens. So it requires cell contact. This may remind you many of these cell selection mechanisms that happen in the immune system, for example. But there are 3 main differences that I would like to stress. One is that the molecules that are doing this type of cell selection mechanism are not involved in the immune system, like these flower proteins. Second one is that there is no specialized cells. So this is among epithelial cells or even among neurons, but there are no specialized cells like in the immune system. So they are just the neighbors that are doing this cell selection. And another thing that I find very intriguing is that relative comparison of these levels of flower loose is very important for the mechanism to work. So essentially, I told you you need cell contact, but cell contact is not enough. So even if you have cell contact between winners and losers or more fit and less fit cells, the probability to die of a loser cell does not increase until you are above 50%. So it's kind of a voting mechanism of a democracy that you need to have more than 50% contact with a fitter cell for this cell to be recognized and eliminated. And the probability to die is maximum when you have a less fit cell completely surrounded by winner cells. We don't know how this mechanism works, but we are very interested in finding out how the cells compare these levels of flower loose. So coming to this, there's very little that we know about how this mechanism works, and I'm sure technologies developed by Janilia and others would be maybe very helpful. One thing, for example, I would like very much to know how to do is to combine live imaging with spatial transcriptomics so that we can select one of these cells in a particular point of geography and time, and then do transcriptomes on that particular cell, as if we will just pick with a robot this particular cell and do the transcriptomics. I will not tell much about the physiological role of this pathway, but essentially what happens to the animal if you don't have this pathway activated that allows you to recognize these less fit cells. What happens essentially is the organism starts to accumulate these viable but suboptimal cells. And the consequence is that you have accelerated aging during adulthood and developmental malformations during embryonic development. And another idea that I wanted to throw here is that I think many of the diseases that afflict us as humans are actually social conflicts of cells. Like basically, that multicellularity still have not found a perfect way to solve social conflicts. For example, this, as I told you, if this mechanism doesn't work, you will have accelerated aging. And if it works too well, you actually can have a very aggressive cancer. And this is just a recent example, a paper that just appeared yesterday, where we were looking in humans at the levels of this flower loose in different tissues. This is the lung. And you can see that there is a lot of dispersion, but as the person's age, there's an increase in the tissues of this flower loose expression, suggesting that actually this correlates with the fitness of the organ and the fitness of the person. But as I said, there's a lot of dispersion, and you can see it also here. But then this dispersion disappears if you check for the clinical history of these people. For example, if you look if they have hypertension, obesity, lung disease, diabetes, or cardiovascular disease, you can see that if they are disease-free, the levels are low. And as you have more and more of these diseases, until the maximum poor people that have 4 of these diseases, you have highest levels of flower loose. And we were doing these experiments during the pandemic, and therefore we decided to explore, because one of the, as you know, one of the mysteries of the COVID is that you can have people that's very susceptible to the infection, and people that's is totally asymptomatic. So you can have death or totally asymptomatic. So because we had this new marker that seemed to correlate with age, and age is one of the markers or one of the signals that a risk factor for death, then we decided to look what's the correlation between death from COVID and the expression of loose in the tissues. And you can see here, these are the people that died. In red, these are the people that is hospitalized. And mostly independently of the age, what you see is that the people that died from COVID and the people that's hospitalized even also correlates with the levels of how human flower loose expression. Even better, there's a better correlation than with age. So suggesting that maybe these tissue fitness markers could be used to stratify people for risk in this and other diseases. So this is the people that work in the lab and do the work and funding, and this is where we are located. Thank you very much.
Thank you so much, Eduardo. I'm just going to quickly throw out a question. Can you tell us what kind of protein this flower protein is? Is it a membrane-bound receptor?
Yeah.
If it's secreted, what kind of
it's a transmembrane protein, so it's a plasma membrane protein. It's a very short protein, and there were some reports originally that this was a calcium channel. For sure, it's not a calcium channel in humans and in mouse. It could be in Drosophila, but even that calcium channel function is not required for this. So we think it's just a very small protein that is recognized. Probably, I would call it a ligand, a transmembrane ligand that is probably recognized by receptors we don't really know yet. I would call it a transmembrane ligand. Yeah.
A question by Hernan Lopez Shear. Edward, do you see evidence of heterotypic cell selection or elimination happening only among cells of the same class?
Most of the time it's among equal cells, so it's neuron with neuron or epithelia with epithelia or stem cell with a stem cell. We have found that some tumors actually are able to eliminate fibroblasts. So some epithelial tumors can eliminate fibroblasts, but I don't know how physiological it is, but this happens in pathological conditions. But we don't have any example of different cell types competing with each other or eliminating each other. And it makes sense if you want, because most of the times you need to replace that cell with someone else, right? So it makes sense that you are comparing apples with apples and pears with pears.
And if you could just quit sharing so that I can see whether there's any panelist questions. In the meantime, I'm going to raise the question from Gaurav Bardway. Are there any cases where fit cells get eliminated instead of the unfit cells?
Sorry, can you repeat it? I was doing something.
Yeah,
I
sorry,
didn't
I was having you do.
Yeah. I cannot do two things at the same time. Sorry.
You can't shoot your basketball into that hoop while we're talking. So are there any cases where fit cells get eliminated instead of unfit?
Yeah, so it depends how you define, but there is cases where the good cell is eliminated. That's, for example, during cancer. So in cancer growth, we have seen that actually these tumors abuse this mechanism to destroy the neighboring tissue and invade. It's an invasion mechanism, local invasion mechanism. So they grow by destroying the normal tissue. So in that case, one could argue that the bad cell is abusing this mechanism to kill the good cell or the beneficial cell.
I'm going to take two more questions. One is from Prabh Gupta. Is survival proliferation by flower recognition an active mechanism? If a cell does not express any flower protein on its surface, does it survive or perish?
Right, that's a good question. Actually, if they don't express anything, they are also eliminated. So if they have to show, that's why probably there are two proteins, they have to show the Wnt isoform. If they don't, they are also eliminated. They are recognized as not correct, not doing well, and are eliminated as well. Yeah.
And the question from Merlin Stapps: where and when did the flower system evolve?
Yeah, that's again a good question. So all animals seem to have it. I don't know if unicellular ancestors of animals have it. Would be easy to check, probably. I remember we once checked with some experts in trichoplax, and there's something similar also. So I would ask you, how I would see it is that I would be very surprised if it's not in all animals, and it's probably something that you really need to have in order to survive well. Yeah.
And Buzzbomb, final question. What about after divisions are over? How do cells suppress killing neighbors in tissues that are, quote, finished?
Yeah, so that's again also a good question. So in the brain, this system works very strongly. Actually, is one of the tissues where it's most important. And of course, in neurons, I mean. So these neurons are not dividing. So what happens is that the neurons are actually comparing, probably at the level of the axons, comparing the levels of these levels of these flower proteins, and then one neuron actually decides, or a group of neurons decide to kill the other neuron, and then there is no really replacement, but we think that there is compensation probably at the new growth of dendrite and so, but we don't really know what happens afterwards. That's a good question, because it's less easy to see. How can you compensate for the loss of a neuron? But it is clearly beneficial, this we know, because it's clearly beneficial to eliminate some neurons, especially hyperactive neurons. Sometimes I make the analogy, if we are now talking here and someone will start shouting, would be great to have more people joining, but if someone starts playing music or shouting, we will not be able to communicate with each other. So there are particular neurons that is better to get rid of, especially hyperactive.
Unfortunately, I can't. We don't have time for the further questions, but there's a whole list of them. Eduardo, please go in and type your answer. But we can also revisit these questions in our discussion session. Hello, everybody, and welcome back to session number 1 in our 4D cellular physiology workshop that's focused on the evolution of multicellularity. We're now going to have 2 invited speakers from our abstract submissions, and then we will move to a discussion. So let's start with Sarah Guest. She's from UC Davis. Sarah, I invite you to share your screen, present your work.
Hi, thank you. Okay. Can we see that?
Yes.
Awesome. Oops, sorry, just moving some things around. Okay. Hi, I'm Sarah Guest. I'm a grad student in the Dawson Lab at UC Davis. And thanks for tuning in to hear about branch morphogenesis in the aggregatively multicellular rhizarian amoeba called Philareta ramosa. Is it going to go forward? No? Hello? Oh, shoot, it's not working. I cannot proceed. Why aren't you proceeding?
Can you try clicking on your keyboard instead of the mouse?
Yeah, neither of them. Oh, there we go. Okay, thank you. It didn't work the first time, but that time it did for whatever reason. Awesome. Okay, so we know that multicellularity has evolved multiple times throughout the eukaryotes, and multicellular organisms can grow to become have larger sizes and can develop like a spatial differentiation with like tissues. And this allows the organism to be able to interact with its environment more efficiently. The accompanying morphological complexity that comes with multicellularity is an emergent property, and this is a requirement to be able to organize those cells into their corresponding functions. Aggregated multicellularity is one subset of development of multicellular life, in which individual cells will aggregate together, and then they'll grow as a single organism. So this form of multicellularity has also evolved multiple times throughout the tree, marked with stars, and it can actually show the change in shape and behavior from the individual cell stage to the multicellular life stages almost like directly. So in this case, if the aggregate actually fuses their membranes, it'll form a syncytium that's multinucleate and has all the cellular components shared across the contiguous cytoplasm. But how do eukaryotes generate these complex and diverse morphologies? So given the basic building blocks that we might expect any eukaryotic cell to possess, how are these like great complex structures and diversity possible? Do they use all new proteins or with new pathways to make new structures, or are they using the same conserved components and employing a new recipe? So I like to think of this analogy of baking. Given that everyone might have all these ingredients in their kitchen already, the diversity of desserts that are possible really depends on the order of operations or the recipe that goes into it. So that's kind of one of those governing rules for this emergent property. With this in mind, a common shape that's seen in multicellularity or multicellular organisms oftentimes is this branch morphology. So branch morphogenesis is one visually obvious readout of spatial differentiation and emergent complexity. And this commonality or convergence arises due to similar problems that the organism might have to overcome, which leads to a similar shape with often similar function. So branching usually involves conserved cytosol proteins, including the microtubules and actin, to drive their morphology. So the Rhizaria are one of the major eukaryotic supergroups that include a lot of branched syncytial forms. The group itself is as diverse as metazoa, and they can grow to be macroscopic sizes like the Caecinophytophore here, which can be the size of a football. They also generate complex shapes, and many of them form tests like these that are used actually in the fossil record. There's only 3 complete Rhizarian genomes and no real working cell biomodels. Many Rhizarian amoeboid protists have this characteristic branching and anastomosing pseudopodia, including the one that I work on, Philareta ramosa. So I isolated it back in 2014 from mud collected from the brackish Sipwasset Marsh near Woods Hole, Massachusetts. We were able to sequence its genome in PacBio and found a lengthy ingredient list of all the conserved cytoskeletal proteins of interest. Since then, I've worked to characterize its life cycle and network development. So first, from the fleeting unicellular stage, individual amoeba will aggregate and fuse to form syncytial fragments. Here we can see that one amoeba is quickly joining the syncytium, while others are still moving around, migrating and eating in this bacteria-rich environment. We can alternatively push an established syncytium through like a 5-micron sieve and get single nuclear fragments that also aggregate and fuse back together. Syncytial fragments will then fuse and grow together, reorganizing their components. And these branches are also capable of autofusion. So through their dynamic growth and reorganization, they'll end up forming this syncytium that branches out as it grows and responds to the environment. Through these autofusion events, the branches form a looped contiguous network with active pseudopodia at the periphery and branch maintenance towards the center. Over time, the syncytium can recognize and grow towards others nearby, And then this will ultimately allow it to cover a surface area of multiple centimeters with its reticulated network. Through certain stress conditions like UV exposure or starvation, the syncytium will begin to form these hubs into which it shuttles all of its cellular components and organelles to be able to make macrocysts, which can then disperse and colonize new areas when favorable conditions arise. Like I said, it is syncytial. So depending on their size, Philareta can contain thousands of nuclei, which move rapidly and bidirectionally throughout the branches of the syncytial network. By transporting their nuclei and other organelles across large distances, the syncytium is also able to adjust and react to environmental cues and maintain that karyoplasmic ratio. We know that they're maintaining this transport and traveling throughout the branches along microtubules because of their reduced transport rates when we treat with the microtubule drug nicotazole and with the dynein inhibitor cilabrevin. So let's look again at that branch morphology as a type of phenotypic convergence of very distantly related organisms. The patterns that we see in Philareta are really similar to those like in neurons and other arborized cell types. So for example, the similarity of morphology is really striking between this radially symmetrical syncytial branch network and the oligodendrocyte here on the lower panel. So the pseudopodia extending from branches of the syncytia also appear to follow similar rules that govern like neuronal arborization and branch formation. This allows us to draw some parallels. So taking what we know from other eukaryotic systems, we can formulate a working model of how the branch network is formed. Branch initiation with pseudopodia and that elongation and eventual fusion or anastomosis is what leads to these loops. And then through repetition of those simple rules, we're able to get an increase in complexity. We know that pseudopodia are driven by actin polymerization, but these pseudopodia are largely transient. So polymerizing microtubules captured in neuronal growth cones can work to stabilize the actin protrusions, which then appears to be the case in Philareta as well. Some, but not all, of the philopodia will be filled in with microtubules, as we can see here in pink. So the overlay shows that only some of them are proliferated. Maintained anastomoses, like this looped network here, are stabilized by longitudinal microtubules, which extend throughout the reticulated network. And then new branches, as seen in this square and this square, you can see that they are able to be filled in eventually with microtubules in that process. And so this allows for a further dynamic interaction and maintenance of the existing network because it can still change the internal topology. Points of anastomosis create these loops in the syncytium, and stabilizing microtubules that traverse multiple branches can allow for long-range transport of those organelles and nuclei of the syncytium. And so the nodes of intersecting branches can become locations for potentially parallel and anti-parallel microtubule bundling. The syncytia also make lamellipodia and philopodia, right? So the network branches are stabilized with when actin and microtubules interact. So we can see that there is a normal curvature in the microtubules, and then when they become captured by philopodia, they become straightened. And this is also remarkably similar to the interactions that are governing neuronal growth cones, where microtubule curvature prior to capture by F-actin filaments can be seen. And then also in neurons, this is pretty tightly regulated to be able to govern which direction the axon head is going to be turning during development. So given the similarities between our neuronal morphogenesis and Philareta's network development, we can see that we can actually apply different similar tools to be able to measure the dimensionality of the branch network. So Shull analysis quantifies the branch intersections across radial distances. And so then we end up with a peak like this. And we can do the same with Philareta, in which we can see a similar kind of morphology. And this gives us an idea of the topology over time, the branch density, and the rate of growth. So in doing this, if I can treat syncytia with cytoskeletal drugs. And in this case, I'm treating with nicotazole. So here we have a syncytium growing out for 20 minutes. And we can see the normal active pseudopodia around the periphery. And then when we treat with nicotazole, the peripheral actin is still being able to make new pseudopodia, but no microtubule stabilization is occurring in the center. And so when we look at the Shull profiles, we can see that with normal pretreatment growth, there is an extension, but then with the nicotazole treatment, there's actually a loss in the internal topology. Similarly, we can treat for actin polymerization with trunculin. And so we can see that there's normal actin turnover here with the pseudopodia And yet when we treat with latrunculin, the actin seems to cease. There's no new pseudopodia being made, even though there's still branch transport happening across the syncytium. So again, comparison with shoal growth, we can see that there are prior to treatment, new pseudopodia being made in extension at that periphery, but after treatment, it's essentially been seized. So comparison of shoal profiles between each drug treatment allows for the visualization and measurement of changes in the branch number and distribution over time. The changes in branch distance to the periphery shows the amount that it's actually expanded, and then we can see with how both drug treatments have affected those rates. The changes in the area under each curve show the change in branch number and internal density of the syncytia that's being covered. So both of these measurements are affected by cytoskeletal drug treatments. Overall, Philaretus syncytia develops a complex morphology as an emergent property of its multicellular life stage. Using conserved cytoskeletal proteins and their interactions, it generates a branch network in a mechanism similar to how neuronal arborization happens. We can affect this network, affect how the network grows with drugs, but genetics really allow a more thorough understanding. Thanks for listening, and I'd be happy to take any questions.
Vivian Chu is asking, do you observe any retraction events, and how does it occur relative to branching events?
Retraction events.
Right.
Oh, okay. So when the syncytium is reorganizing itself, and if it's retracting one branch in order to kind of reconserve or reorganize those components, it can then allocate and increase its branch degree or rate in a different direction for sure.
Okay. Okay. And are they coordinated is the question.
I haven't looked at that specifically with being able to measure it, but I would believe like definitely that once it's being able to coordinate and take up those components, it can reallocate them rather than having to grow them completely over.
And Yuri Pishugin says, fantastic organism. As I understand, it grows as solitary cells at the beginning, and then they merge into the network-like structure. Yes. Do you know what is the ecological advantage to transform from isolate amoebas to the merged state?
I don't know exactly what they're doing in the environment. I do know, I mean, we're growing them on like a plastic dish surface that's one-dimensional, or I guess technically 2D. It's a plane rather than a 3D matrix like how it grows in the sediment. My guess would be based on how, when I culture them, they grow as an individual amoeba when they're in a much denser nutrient environment. So on a plate, when you've got a lawn of bacteria, that's when they're most likely to form the amoeboid stage. And then when you transfer them to liquid, it's a lot more dilute so that you can actually image them and not have a bunch of bacteria being shown. And that's when they seem to change over into that syncytial stage. So that kind of gets to the cue maybe of they start fusing and becoming a syncytial form when they are in a lower nutrient environment, because maybe that's a little bit more efficient for them to grow in that manner, would be my guess.
A different question is from Buzz Baum. Sarah, great talk, beautiful pictures. If you UV damage individual nuclei or mitochondria, can you see isolation, competition, or clearance?
Okay, that's a little bit tricky. So I do imaging with DAPI and Hoechst, and they seem to be a little bit stressed out by it, but they typically will just mostly go into their hubs. I haven't been able to actually see whether or not they're excluding certain nuclei because of damage or anything like that. I haven't observed that specifically happening, but that would be a really interesting thing to pursue.
And related to that question, do you have any idea what the nuclei are doing as they move around? Does the Golgi apparatus, for instance, which is the secretory organelle, follow the Golgi? Do these nuclei define sites of localized secretion or what?
We absolutely
secreting. This thing is growing.
I love this question. Yeah, I love this question. So we can speculate. We think that maybe they have different transcriptional jobs, and so they are kind of commuting to their different locations based on what the network needs for its dynamic interaction. But we haven't been able to do any sort of single nuclear transcriptomics on it yet, or looking at that specifically. As far as where the Golgi are, I mean, we know that the other organelles, like I've seen mitochondria and lysosomes trafficking as well, at slightly different rates from what the nuclei transport occurs at, but I'm not exactly sure where, how, why they're doing all of it, but definitely something that I want to look at in the future.
Okay, cool. And then Renski Rumrans says, these organisms are amazing. I was wondering about the comparison to neurons though. Do neurons also form these closed loops?
So not all neurons do. Some of them will create these anastomoses, but they usually don't maintain them. They'll kind of zipper them closed with the microtubule bundling and membrane pressure and things like that. However, recently it was published by a, I think it was Pavel Burkhardt's group, actually published on ctenophore neurons. And so comb jelly neuron systems do make a neuron net, and they maintain their loops in a similar manner.
This is one last question among many. This is Amaya Duden. Is it possible that the network growth is also dependent on nuclear division transport? When nicotazole is used, nuclei do not divide anymore, and that may lead to your phenotype. Is that the case?
That nuclei don't divide because they're not able to make a spindle.
Is the network growth dependent on nuclear division or transport?
I see.
More precisely address that question.
I haven't looked at that. I mean, we grow them without the presence of nicotazole, and either way, yeah, that's a really good question. I'm not sure how to answer it, actually.
Alex has a follow-up. Can you recover cells from nicotazole and latrunculin treatment?
Yes, you can. You can wash it out, and they'll be just fine. They'll recover in a matter of minutes after that. Okay. Yeah. Sorry about that. I wish I could answer that questions.
It's really exciting. Go in and take a look at them, and we can hopefully revisit some of them. But we need to move on. Our next speaker is Tatiana Mararova from NINDS, NIH. Tatiana.
Hi, hello everybody. Thanks for inviting me. And today I would like to talk about macrophage-like cells and their evolution. Sorry. Sorry, I cannot move. Oh, here it is. Sorry. So macrophage-like cells are present in almost all metazoan phyla, except for few groups. They have different names, but have the same appearance and functions. These are mesenchymal motile cells involved in innate immunity and wound healing. However, whether placozoa have this cell type remains unknown. Therefore, the purpose of our work was to check whether placozoans have macrophage-like cells as other animals. We worked on Trichoplax adherens, the most well-studied species of placozoans. These are flat, pancake-shaped animals using mucociliary locomotion to glide, and they feed externally, unlike other animals. They consist of 6 morphological cell types: ciliated dorsal epithelial cells and ventral epithelial cells, digestive lipophils involved in digestion, gland cells secreting neuropeptides and mucus, and 2 types of non-epithelial cells, which are crystal cells responsible for gravity reception and fiber cells with long processes. So among these 6 cell types, fiber cells seem to be the most promising candidate to be a macrophage. So what are fiber cells of trichoplax? These are large cells with multiple processes scattered over the ventral epithelium. Occasionally, they are interconnected by specific plug junctions. At EM level, they can be recognized by a mitochondrial complex and a large inclusion. Their processes filled with microtubules and actin filaments. They bear rickettsial bacterial endosymbionts in their endoplasmic reticulum, and they've been reported to have food particles inside, but how they got inside remain unknown. Therefore, we further test whether fiber cells are placazone macrophage. It appeared that fiber cells are able to phagocytose dead cells and cell debris. Here on ultra-thin sections, a fiber cell extends its multiple processes and in wrap the particle, which presumably is cell debris. Here at higher mag, you can see the material with membranous content and remnants of organelles enwrapped with multiple branches processes of fiber cells. Further, we saw phagosomes within the fiber cells with membranous content similar to that of the cell debris. And moreover, some fiber cells possess even several phagosomes with different degree of homogeneity of their content, which probably reflects consecutive stages of degradation of their content. And here is a crystal cell with a crystal inside. And it became apparent that fiber cells phagocytose crystal cells because occasionally crystals are observed in fiber cells inclusions on ultra-thin sections and in vivo by DIC optics. Apart from cell debris, fiber cells phagocytose bacterial pathogens. To show that, we challenged dissociated cells with heat-killed fluorescent gram-positive and gram-negative bacteria and added also vital dyes for lysosomes and reactive oxygen species typically generated in phagosomes. And in all experiments, bacteria located over the fiber cells were labeled with both vital dyes. By contrast, bacteria outside of fiber cells did not have any labeling with vital dyes. And the fiber cells were the only cell type demonstrating phagocytosis of bacteria. So taken together, fiber cells can phagocytose cell debris and pathogens, and this indicates that they provide innate immunity. Next, we checked how fiber cells respond to wound healing. Trichoplax cut in half heals quickly in less than 1 hour. We fixed stages during wound healing and did immunocytochemistry with the antibody which labels fiber cells. In intact animals, fiber cells lie deep inside and never contact the environment. However, in wounded animals, fiber cells accumulate along the wound where they get aligned and stretched with their processes along the wound. And in this way, they probably contribute to wound shrinking. So taken together, we demonstrated that fiber cells indeed involved in innate immunity and wound healing, and thus can be considered placazone macrophage-like cells. Therefore, each metazone phylum have macrophage-like cells, including placozoa. This means that perhaps the last common ancestor of all metazoan already had a macrophage-like cells. Although multiple origins of this cell type cannot be excluded, especially if we assume the paraphily of sponges. And now the question is how macrophage-like cells evolved. There is a hypothesis that the evolution of cell types precedes the evolution of multicellularity in animals. The first cell types were present as different phenotypes of unicellular organisms in their complex life cycle. Let's take a look at two unicellular organisms which are close relatives to metazoans. They have complex life cycle with transitions from amoeboid phenotype to swimming flagellated phenotype, which sometimes demonstrate tendency to aggregation. We hypothesize that macrophage-like cells evolved from amoeboid phenotype of these organisms, while epithelial cells might have evolved from flagellated, swimming, or aggregated phenotype. Later on, in the first multicellular organism, this epithelial cell might have differentiated into 4 ubiquitously present morphological cell types by division of labor. So here comes a picture of how first cell types might have evolved in the last common ancestor of metazoans. And I would like to thank people involved in this project, my supervisors, and other people from our lab and from elsewhere. And thank you for your attention, and I'm happy to take your questions.
My question, I'll just start off the questions, which is, we've heard a lot about during the presentation, lots of examples of cells that are coming together to create a multicellular system. But there's a lot of animals that basically start with one big cell, an egg cell that gets fertilized, and everything else is just division. These seem to be very different strategies for creating a multicellular system, and I'm just curious as to how you think about that. Basically, multicellularity sort of driven by cells having an adhesive capability versus multicellularity that's just driven by division.
Thanks. Yeah, that's a really interesting topic. And of course, in lots of cases, the multicellularity is driven by cell division, but also there are, cases where the aggregation of cells is the cause of multicellularity. And well, we don't know a lot about placozoans in this respect, and their origin, and it's getting more complicated because we don't know the developmental processes of placozoans. That's a really big problem in understanding on how the cells in these organisms are communicating and controlling each other. Yep.
Manu, since you study this organism, I'd like you to sort of join in the conversation in terms of what you think are sort of some unique strategies that it has in terms of I mean, I thought it was quite interesting that some of the cells inside this placozoa actually are housing endosymbionts as part of their activity. Other thoughts or musings, Manu?
Yeah, I mean, I think first of all, Tatiana, fantastic talk. I have tons of questions for you, but we can also take some of those offline. They can become very particular. But I think, the notion that this would have a mechanism to harbor and control pathogens was almost essential, because you're living in this bacterial environment and you needed. And I think there are very particular pathogens that we've known that they're not able to fight. So this is really an evolutionary battle in some sense, going back to the original question that Iñaki had mentioned about the environment. And unfortunately, the fact that we cannot recapitulate the original environment of these organisms makes for a really big challenge. I think on the comment about this notion of adhesion and free living, there is possibly, we believe, a free living form of placozoans as well, because actually this is how we collect them in the ocean. But we don't have the physical understanding of what that free living form is. So there might be another morphological form that we are completely missing. And again, going back to other than genetics and going back to the notion of environmental sampling, when organisms have completely different phenotypes and some of them are harder to find, it becomes quite challenging. Are we studying the complete life cycle and how much plasticity exists in many of these multicellular systems? So this notion of course, the name of this organism is associated with being a hairy plate, which is sticky, might only be one half of its life cycle, because we do know it exists in a planktonic form.
Yeah.
And we've been trying to get them, and it's been very difficult because they transition extremely quickly from a planktonic form too. So we might be missing half the picture, but that's the reality of studying these fringe organisms.
So let me just throw out some questions from the audience. Koryu Kin is asking, is it possible that fiber cells act not only as macrophages, but also as pluripotent stem cells, like the archaeocytes in sponges?
Yeah, thanks for question. It's really interesting, because not only in sponges, but in many other animals, these mesenchymal cells are indeed stem cells. We don't have a certain answer for that question. However, it doesn't seem like that, because there is a separate pool of cells which seem to be mitotic cells, and the newly differentiated cells do not have any organelles or features of fiber cells, which might indicate that they originated from fiber cells. So yeah, the answer is probably not, but it should be.
And a second question from Ruslan Medzhedov. Great talk. Are these cells, the fiber cells, also responsible for extracellular matrix production?
Yeah, thanks. That's a good question. And another big problem, because according to transcriptomic study, yes, fiber cells express genes for ECM, but on the other hand, placozoans do not have, apparent extracellular matrix. So whether these genes are indeed involved in ECM formation or not, and where is this ECM, is, all these questions are still open.
And then one last, well, I got some other couple questions. This is Nat Clark, wonderful work. Do the fiber cells that move to sites of wounding differentiate into epithelial cells following wound recovery?
Thanks. Very interesting question. And indeed, in sponges and some other animals, the macrophage-like cells behave exactly in this manner. We think that the fiber cells can actively migrate to the site of the wound because we demonstrated that the cells are indeed motile. And since they're getting accumulated at the site of the wound, we presume that they actively migrate there. However, whether they transdifferentiate there and what exact role they play in the wound healing, we unfortunately don't know.
Okay, great. Thank you. This is really exciting work.
Thank you.
Beautiful morphology, super interesting. We're now going to transition into our discussion. I'm going to sort of invite all of the speakers, you'll be coming sort of on screen. We're going to sort of open it up to a broader discussion. So people in the audience, please start putting questions out. But also, I would very much like to hear question answers among the panelists as well. So I'm going to start with sort of a big question related, and actually this comes out of sort of a broader set of questions from the audience, which relates to do you see that there's a sharp boundary between an individual cell within these populations versus the whole cell population, To what extent are individual cells sort of the unit of these systems as opposed to being really subordinate to the larger scale operation of these cells as a group? Any takers on that one?
I loved, I think Kiko used the term superorganism, and
yes.
Thinking about these large trees and plants, and I love that term. I guess maybe one way to just approach the question is to think about, what kind of goal or context, to pin that goal or context, and then what is the minimal functional unit for a particular goal or a particular context, that could perhaps serve if the functional unit is a single cell for certain types of behaviors. It could be a small neighborhood of cells or a niche for other types of behaviors, and then very large aggregates or very large cooperativity between many cells for still other types of behaviors.
So one of the sort of challenges that Janelia is facing with the introduction of this 4D cellular physiology research area is, basically we're moving from cells that are being studied in just tissue culture dishes to cells that are part of whole organs, whole tissues, or whole organism. And I want to ask you all whether you feel, what are the surprises that you anticipate? Each of you have delved deep into very different organisms, ranging from plants to, some of the most simplest multicellular systems on Earth. And each of you have really discovered some new things, some very new biology. To what extent do you think in this 4D cellular physiology context, where we're going to be revisiting cells that we've been studying for years in cover slips, what surprises do you anticipate that will be sort of met and how can we, what are the tools and ways that we can sort of keep our eyes open to what Anako talked about as the invisible. That's we haven't thought about it yet because we haven't started in sort of pushing into that world. Any ideas? I mean, I think, for instance, one of the things that I thought was very interesting is this competition. What's going on? How are cells competing with each other? What's the mechanism by which they're making decisions as to how you fine-tune the organization of a particular multicellular grouping? One of the questions from the Q&As was, and this I think links into sort of physics, what is the biophysical forces that are driving a lot of the systems that you're seeing? I mean, basically it's, on the one hand, there's physics that are putting constraints on the system and driving these systems in different ways, but there's also metabolic characteristics. These systems are moving into different environments where there's more nutrients or less nutrients. Do you see these principles as fundamental characteristics of all of the systems that you're looking at? I see.
Definitely.
Kiko, why don't you? Yeah.
I mean, I think everybody is in agreement. Definitely. Yeah. I don't study the cell system in a plate like a fibroblast or myoblast or iPS stem cells or ES cells, but I'm sure everybody has feeling or observations that in a natural or in natural context of in the organs or tissues or organism, that each cell behaves so much better than in an isolated situation, right? And somebody asked the leaf shape, but they can be bigger or smaller, but then they know exactly what kind of shape that if you're a maple tree, you generate maple leaf, and if you're an oak tree, they generate oak tree. They know exactly, even though their cell types are pretty much the same, they know exactly what kind of shape they make and then adjust the size according to the nutrient availability or the temperature or the climate, so that they can maximize the productivity of the tree as a whole, not just the individual leaf's productivity, because leaf makes nutrients to support the growth of the entire tree, not just the leaf is not just for leaf, right? So, and stomatas are made to adjust the air, which the internal mesophyll tissue do actually make sugar through photosynthesis. So everything is so coordinated, and I feel like animal field has advantage that they can do tissue like cell culture and stuff so well, the drug treatment or everything. But at the end of the day, when we think about the entire system, I do think everybody knows that the cell behaves so much more part of the team rather than when you are culturing cell. And of course, cancer cells is deviating and become selfish in a way, but I think everybody knows, and it's going to be drastically different. And how are we going to put into the context through understanding the different system would be critical. So something that I would like to learn from scientists studying the different system, non-plant system as well.
Exactly.
Jennifer, maybe I can actually give you 2 cents to your question about the 2D or 3-dimensional system. So that we know actually from experiments by many people, including Chris Chen, actually, that if you look at the response of cells actually, for instance, to mechanical cues in 2-dimensional systems and in 3-dimensional systems is very different. Actually, in some cases, it's even the opposite in 3D compared to 2D. And this is only in vitro. It's not even in the embryo, actually, because in the embryo, we have very few approaches actually to study that question. So from a physics perspective, there are many different processes that that are very different in 2 dimensions or in a dish from a 3-dimensional organism. So I'm pretty sure we're going to see many different aspects when we actually are able to really explore them in 3D. The other thing that I wanted to point out is that, and this I'm not going to talk about it tomorrow, but we've been studying also morphogenesis in wall cells, walls meaning yeast, meaning plants, during tip growth. And actually what is very funny is that, well, very interesting actually, is that nearly all of these organisms have a common physical mechanism of growth. However, actually the molecular encoding, the molecular control of that mechanism is different. It's a completely different mechanism that evolves differently in different organisms. But the physics in those systems constrains enormously the morphogenesis process. Whereas in animals, in multicellular organisms, we see kind of the opposite, actually. You could generate a ton of different shapes. It's this combinatorial code, actually, that you can generate essentially what you want, and physics is less constraining. So maybe it's something, actually, that the others that have different organisms actually could chip in their ideas.
If I can.
Oh, sorry. Who?
Lucy.
Lucy, please. And then Manu has a comment. Yeah. Lucy.
Maybe if I could riff on Okur's comment and mechanobiology, mechanotransduction for a little bit more. I think it's true that as biologists, you know, we've become really used to and very comfortable about thinking of the modularity of signaling systems or the modularity of certain cellular functions. You know, so if you're talking about metazoan cells and talking about mechanobiology, we start thinking about actomyosin contractility, for example. But I think what will come out more and more as we study things in a more physiological context is the incredibly different length scales and time scales that these modular cassettes are actually functioning in. We have the same actomyosin contractility, you know, same molecular players that can exert forces on scales of microseconds, and also that play roles, you know, critical roles in events that happen over multiple days in other tissues where cells differentiate. So how does that work? Well, how can you have the same molecular cassettes, molecular players that are exerting their functions over such different timescales? As another example, you know, in Sarah's networks, I noticed, you know, Sarah, when you were comparing your branching networks to a neuron, neuronal arborization, the patterns were so similar, and the cytoskeletal components, cytoskeletal processes are similar, but the length scales were really different. And so, you know, so what plays out there?
Very cool. Manu.
So I wanted to make a comment related to what Inaki had, I think, brought up early on, but also now what JLS, you mentioned about the role of 4DP in this context. I think the idea that we have to be first comfortable with the fact that there is probably 100-fold more or even more of sets of organisms that we don't know about. The pictures that we paint is from what we know. And so how do we, as a systematic approach, appreciate and understand just the enormous biology that we currently don't know? And what systematic approaches would we bring to the table to expand that? It takes, for many of us who work on non-model systems, it takes years and years, 5 to 10 years, just developing the tool to ask that one question. And sometimes being able to systematically build, both local communities that some of the groups have done, but it has its own challenges itself, the amount of effort that goes in that front. I could really see 4DP playing a big role, especially in this context of multicellularity, if we truly value the new discoveries that come out of this. And I think, this is a big question because much of the funding is tied to biomedicine. And then the second comment I wanted to make in the systematic approach, again, going back to the environment, I think we have something to learn from the neuroscience community. They have really used and exploited the notion of virtual reality, to ask a question in a Drosophila, but in a phenomenal environmental context, mice do the same thing. There are many common techniques that can be developed, and technology is at this moment where we could recapitulate certain aspects of the environment. something that we could share amongst each other that doesn't have to be reinvented over and over again, to really ask these challenges questions in context of the environment. Because without the environment, many of these biological discoveries are devoid of the evolutionary context. And it's, for us to truly capture this picture, and I'm wondering, how could we as a community speed that process? Just like genomics has happened, what are the other, without this context of purely, what we need is an omics revolution in physiology. And we cannot do that alone unless we band together in a certain manner. So I'm curious, how do people reflect on that? When will it be that we will start really reaching? Every single organism is hiding thousands of mysteries, and we just have so many. And it kind of makes me sad in the context of COP15, we should mention the biodiversity loss. Microbial organisms are lost much faster and nobody cares. So what are we losing at a speed that which we're not even able to study?
Manu, on that point, do we know how fast these small multicellular organisms are being lost now in terms of climate change and other problems?
Yeah, I think there are certain estimates that are coming out because there are really nice mappings between ecological niches and biodiversity. Yeah. And so those sets of estimates are quite scary. In the context of thinking about even the macroscopic or mesoscopic species, the rate loss is so fast. And sometimes many of these organisms have really specific ecology. One example on a microbial context is the Arctic, Antarctic ice. The ice harbors gazillion number of protists and has a very specific ecological niche, which is this interface between the seawater and ice. The faster the ice we're actually losing an environment that's completely unique. That's just one example of the fastest, biological niche that we're losing, but it's the microscopic context is not at the front and center of some of the ecological perspective where many of the biological mysteries might actually be hiding there.
Zev, do you have some comments related to that or other areas?
I was going to move in a slightly different direction, so maybe I can wait a little bit till you guys are on.
Okay. Inaka, did you want to say something following up on Manu's comments?
Yeah,
or something new
about this systematic analysis of trying to unravel everything that we don't know. That, of course, is very difficult, right? But there is a way in which you can approach that, and it's ranking what is the most important things at each moment, and you can define some, for example, in the origin of multicellularity or transition unicellular multicellular. There are some organisms that you can decide previously that are, very important to anchor in the most deeper knowledge. Of course, once you have those, it's better, it's easier to compare whenever you find in other organisms. So that's one way, you define something. Another way is to look which new organisms you can find, and then also rank them also whether they could be anchors, so they don't need to be anchors. So that, I think that, there is that, and of course we need to focus on the ones that are more important. Some communities fly by themselves, so you don't need to worry too much, and they will always be at the foreground, and they will always bring the technology advances that later on we will adapt. But we need to do a decision in some of these organisms to go deeper and use them as an anchor for later analysis in other ones. So that's one way. And then coming back to the 4D, I don't, if I was, there is nothing that I really expect that there will be unexpected, because then it wouldn't be unexpected. But if you go to more complex things from 2D to 4D, you will, of course, it's the complexity of things that you bring in, in the environment, in the context of the cell, in the cooperation, in the coordination, and so on and so on. It increases significantly. And then you need to have the way to analyze that, and also to interpret the data that you have, and to be, it will get more complex. So I know in some fields they prefer more simple things. I think it's useful to have more simple models, but here it will be more complex, at the same time more natural. Usually the more natural, the more complex it is, and I think we need to be aware of that.
Great. Zev, do you want to comment?
Yeah, I mean, the question comment that I had was moving in a slightly different direction, was based upon, in Sarah's talk, I was really thinking about these cells that would join these nuclei, if you will, that would join the larger organism. And I was wondering, what were the limits of that? Were there mechanisms that this organism had to exclude certain classes of cells that might be genetically different or otherwise? And that got me thinking a little bit more about some of the other presentations and this sort of intricate balance between cooperation, cells coming together, and competition, keeping cells away. We heard that in Trichoplex, et cetera. So I guess what I was wondering is, can we think about this a little bit through the lens of function and the importance of maintaining function, whether at the evolutionary scale or even at the level of an individual tissue? And I was just sort of curious what the speakers thought about this question as it pertains to their own work and others.
Lucy, you want to start? Yeah.
Yeah, I can start. I love thinking about these questions about do we add on or do we take off and shed? And one thing that Zev, where physiological function or physiological context regulates that balance in the fly gut is that we find, the fly gut's always shedding cells at a basal steady state rate during renewal. But if we take food away from a fly, and so now the fly is starved, the fly gut starts to shed cells at a massive rate. And normally during steady state turnover, all the cells that are being shed are apoptotic. We see them, they become apoptotic in the epithelium, and then they get extruded. But in the fly gut, when we take food away and the gut now, it contracts because there's nothing in its lumen. Now there's a massive activation of live cell extrusions. That actually reminds me, I think if Buzz Baum is here, reminds me of some of the work that he's done, showing a role for mechanical compression in developing tissues in driving these live cell extrusions. So there's a case where it's the same cells, the same tissues, and a change in physiological context that is influencing, whether cells are being breaking out or not. And this can, repeat over and over again as feed fast cycles throughout the fly's lifetime.
Great. Sarah?
Yeah, my comment on this has to do with my system, and I guess it's a little bit more broad, but it kind of answers a couple of the questions or goes along a couple of the questions that I received. So when the organism, when Philareta, for example, is faced with a different situation than it grew in, it will interact in a dynamic way with its environment. So when you starve it, it does interact too, where it simplifies its branches down so that it can still maintain like the really basic function. So instead of having a lot of intersections with its loops, it's going to have fewer intersections with larger distances between them. So then you end up with a more simple looking topology of the overall network. On the other hand, when you add in nutrients, it starts getting more and more branches, and it even at that point can start budding off individual amoeba again. So it's like a different, and I didn't have time to be able to show those sorts of movies, but it has a different format of like its response when it's given a favorable environment, when it's already in this syncytial form too. And I feel like that's...
Did the amoeba bud off? I was curious about this earlier too. Did the amoeba bud off individually then, or do you ever see like a synchronous deaggregation of the whole syncytium?
I haven't seen a synchronous deaggregation where like the whole thing just disperses itself. But I have seen like a pseudopod will crawl itself away kind of thing into its own little amoeba. But I mean, yeah, that's definitely one of those things, right, where it's interacting with its environment and recognizing a certain change that has to happen, and it can kind of switch back and forth between that decision making of stay multicellular or not. But yeah, I think that's something that if you can get at a question with your system of whether or not you can modify the environment and get a direct readout of what happens morphologically or characteristically with those cells, that's really like
let's go ahead
a super helpful system to be able to work on, right? So, yeah.
We're coming close to the end of the time, but Keiko,
Allison, do you see? Allison, do you have other?
Yeah, hold on. Let me get rid of the hide voting meeting tools.
Short, kind of like a concluding comments through what I felt.
Now.
And I agree with everybody that the cell compatibility or the cooperation or competition, like when to betray or when to cooperate, I think it's fundamental survival strategy of any scale
Fred,
of the biological
Eric, anybody
system,
who
right?
wants
So I
to
think
test,
that's
you could
really
test
the
their screens.
interesting
I don't know, Janine, how much time?
exciting talk in this session.
Like
and in plants, I said plants can be fused and grafting and wide variety, but there are compatible
Janine, we still have time, right, or no?
Combinations that we still don't know what's the basis, and scientists found that one species, Nicotiana benthamiana, is kind of like a universal donor. So if you put that plant in between, then the two disparate species that are otherwise incompatible can join, like a three-way jointing. So that's giving some tools. And how we can address this non-modal system, I think there are two things. That some consortium in the plants, there was a 1,000 plant transcriptome initiative.
So you could
that
choose
just
any of the
like
options.
Species of completely different phylogenetic status. That gave us a lot of tools to explore the model system, as well as recent CRISPR-based implants. We use the plant virus to do some sort of CRISPR-based genetic track. So that made a genetic manipulation of otherwise non-models, genetically untractable non-model system possible. So I think though the wide variety of genome sequencing of many different organisms and developing sort of CRISPR-based technology that can allow genetic manipulation of non-model system, they all have to come together to really understand the broader system. And when it comes to funding, I think it's so critical. We identify small groups of scientists or small labs
Karina, if you want to test in the last seconds left,
like Sarah's system, like weird, strange creatures that has enormous potential, and the funding mechanism such that to allow those small groups studying the small weird creature.
So Janine, I'm giving the control back to you. So whenever it's time, you can let us know when to start.
Or something like that. And I really hope Janelia look into that, because those small creatures could give rise to enormous funding that changes our viewpoint.
Actually, we're right about 1:45 right now. So why don't we go ahead and get started with the session, Manu?
So that's what I.
Thank you so much.
Okay, welcome everybody. Welcome back. Not sure how many of you are actually back or still taking a break. That's okay. I think this session is going to be focused a little bit on frameworks. Some of those frameworks will be embedded in mathematical thinking, physical thinking, but also philosophical approaches to this grand challenge that we have in front of us. I think in the first sets of sessions, you all got to see how exciting many of these discoveries can be. And my hope is in this second session, we'll try to share approaches where how to really put in systematic order those sets of exciting discoveries in a larger context of what we think about evolution of multicellularity. So with that brief intro to the session, I'm going to invite Wenying Xiao from University College London to give the first talk.
So I'm Wenying Xiao. I want to discuss forcing a transition in the unit of selection. What is a unit of selection? It is a unit on which selection acts. So unit of selection is an entity with a trait, and an entity should be able to reproduce. And the traits of the offspring should bear some resemblance to the trait of the parent. And I call this heritability of the trait. Furthermore, there should be variations among these traits so that selection can act on the trait. Example of unit of selection include self-replicating nucleic acid, a cell, or a community of cells. The history of life is characterized by transitions in the unit of selection from lower level unit to higher level unit. The theme of this workshop, the transition from unicellularity to multicellularity, is one such transition, because we have a shift in the unit of evolution from a unicellular organism to a multicellular organism. And the multicellular volvox has somatic cells that allow the organism to swim toward nutrients and light. It also has germ-line cells that are responsible for reproduction. So here we see the division of labor. Another example of such transition can be seen when self-replicating nucleic acids are strung together to form a genome. And in all these cases, the lower level components must show restrained proliferation so that proper proportion of the components can be maintained. But this restrained proliferation can be broken by cheaters. Cheaters can overproliferate. For example, this is a mutant volvox where you can see the small somatic cells now are dividing. And in genomes, there are transposons who divide more frequently than the genome. And that's why evolutionary biologists call this class of problems multi-level selection, because selection acts on these higher level collectives and at the same time acting on the lower level components. And I want to discuss in this framework of multi-level selection an application, which is artificial selection of multi-species microbial communities. Multi-species microbial communities can often perform functions that are displayed by the whole communities, but not member species alone. And there are many such examples. And one example is the degradation of cellulose in the form of paper. This species cannot do the degradation, and neither can this species. But two species together can degrade the cellulose. And so degradation of cellulose is an example of community function because it's an activity displayed by whole communities, but not member species alone. So now let's imagine we have a newborn community, which I will shorthand as newborn. It has two species. And then we allow the community to mature. By maturation, I mean we allow cells to grow. And the duration of maturation is determined by the experimentalist. At the end of maturation, we obtain adult community or adult. And so suppose this adult has this community function indicated by the shade of this tube. And community function can range from low to high. And suppose that we only have communities with low functions. How might we improve it to high function? Now, there are many ways of doing this, to achieve this. But one possible way is to do artificial selection of whole communities. That is, we use a community as a level of, as a unit of selection. And the advantage of doing this is that we might be able to improve community function without understanding how species interact to generate this community function. Conversely, once we have obtained communities with high function, by comparing DNA sequences of evolved communities against the ancestral communities, we might be able to form a good hypothesis about the mechanisms of community function. So the procedures are following. We start with a population of newborn communities, and we allow them to mature. And during this process, cells will proliferate, different species will interact, and mutations will arise. And then at the end of maturation, we obtain adults, and we'll measure the community function, and then we'll rank them according to from low to high. And community function can be, so for example, the degradation of cellulose or the production of some compound. So after we rank the community function, we then choose adults with high functions to reproduce. And the reproduction occurs when we split each adult into multiple newborn communities for the next cycle. So now I want us to think through this entire process in the three points that I just discussed. Shift in the unit of selection. This seems to be the case because now the unit of selection and the unit of reproduction is a community instead of cells. And I'll return back to this point in the next slide. Division of labor, of course, there are division of labor. In fact, that is the reason why we're doing this experiment. Okay, now restrained proliferation of components to maintain proper proportion. So let's consider a case where this oval cells would divide faster than its partner species, the rod-shaped species. We can imagine that there might be a cooperator, you could evolve a cooperator mutants that actually divide slower. And this restrained proliferation can allow the two species, the oval and the rod, to coexist. On the other hand, we can also imagine children mutants that show unrestrained proliferation. And this will divide even faster than ancestor, and that will drive partner to extinction even faster. So now let's think through this process. Right? During this maturation process, those cells are competing, the cooperator, the ancestor cheaters are competing against each other. Who will win? Of course, cheaters will win because they divide the fastest. So this maturation process favors cheaters. And what about at the level when we select communities? So note, if a community is dominated by cheaters, the partners are present at very low frequency or even driven to extinction, and then community function will be low. So cheaters will be disfavored at this level, and this favors cooperators that show restrained proliferation. So we see precisely this multi-level selection at play again. You have inter-community selection and intra-community selection. And in fact, we have shown, we and others have shown that if you do the experiment properly, you could in fact evolve restrained proliferation. So I want to return back to community as a unit of selection. So is community a unit of reproduction and selection? Well, with the help of an experimentalist, that can be done. So it's assisted reproduction and selection. What about the variation in community function? So then one would need to ask, what contributes to community function? One could write down a series of differential equations. So community function is, would depend on initial conditions and the parameter values. And the parameters include the phenotypes of the cells, including how they interact with each other. So variations in community function can be introduced if different communities would acquire different mutations. But I want to remind you that one type of mutation that is for sure going to arise are cheater mutants. And those would show up in communities. And so if a community, a newborn community, starts with lots of cells, then the cheaters are almost inevitably present at very, very early on. So then they would all take over, and then there'll be no variation among adults. And then indeed, when we look at fraction cheaters as a function of time, and each colored line means a different community, we see that if we start with 100 total cells per newborn, even though cheater always increases in frequency, there is variation among communities. In contrast, if we had started with many more total cells per newborn, there's very little variation among communities, and the cheaters just take over very deterministically. So that means, to achieve variation in community function, newborn communities should not be too large to ensure variations among communities. What about heritability in community function? And what is heritability? Heritability is a similarity between the parent and the offspring. So if the parent community function is strong, dark, and so on average, the offspring adult community functions should also be on average large. In contrast, if the parent is weak, and then on average, the offspring should also be weak. Now, if we think about it, so these two have to be similar. So then this step, right? So these newborns should look like this, and these ones should look like this. But to ensure that newborns look like the adults, then we want newborn size to not be too small. Otherwise, stochastic fluctuations can maybe we might lose a species during this splitting process. At the same time, the maturation time should not be too long, right? Because if it had been too long, then cheaters will take over in every single community, and then the offspring would not look like parent. And indeed, if we look at fraction of cheaters as a function of time, so if we have short time, we see that the cheaters take over in some, but in at least some of them, the offspring looks like parent. Whereas for a long time, all communities are dominated by cheaters, and so the offspring would not look like parent. So to conclude my talk, I would like to list a few open questions. First, how do communities evolve under the influence of intra and inter-community selection? What determines the variation and heritability of community function? How might experimental variables affect the variation and heritability of community function? And there are many experimental variables, including newborn community size and maturation time, which we talked about, the total number of communities under selection, and the percent communities chosen for reproduction, and so on. And that's why it's critical to combine analytical calculations with computer simulations with experiments. And the reason is because there's so many variables, and these experiments are highly non-trivial. So to just probe them blindly to figure out the best way may not be the best strategy. And computer simulations can allow us to figure out realistically how experiments might look like if we had performed in certain ways. But analytical calculations will actually show us why we are getting the simulation results we're seeing. So with this, I want to also thank people who contributed to this work and the funding source. So with that, I'm ready to take questions. Thank you.
Thanks, Wenying. We have maybe 3-4 minutes for questions. I'll start with one that's in the Q&A already from Rensky. This is a fascinating protocol, Wenying. Do you apply selection for cellulose degradation or any other function throughout community maturation or only at the end?
So we do that at the end of it, because actually measuring community functioning is not a trivial process either. And then, so that raises a very good question about how long the maturation time should be. When would you assay that, right? And as I showed that the maturation time cannot be too long, but at the same time, it cannot be too short either. If it's too short, then there are no opportunities for mutations to arise. So you can see this tension, right? So because the newborn size must be not too large, and it must not be too small. The maturation time must not be too large or too small. So that's why quantitative analysis is critical in this type of experiments.
And then there is a follow-up from Rensky again. Also, do you observe a correlation between amount of cheaters and community fitness?
So if community fitness is defined as a function that requires cheaters to not be there, then if a community is dominated by cheaters, then by definition, the community is not fit when you do inter-community selection.
And then I think I wanted to ask a higher level question in this framework. I think as you highlighted the value of thinking about multiple scales of selection, could you comment on the challenges associated with mathematical frameworks that have to go, because you now have to account for multi-scale physics, all the way from what biological processes might be at play in a single organism to literally some of the things that are happening metabolically in this community. So, where do you draw the line in terms of reduced order models or actually capturing as much complexity as possible, even in these toy systems? I'm just curious on the theoretical framework, where do you lie? Where would you like to be, say, 5 years from now?
Yeah, so I think that's an excellent question. And so the hope is the following, right? So we would start with maybe simpler communities where the number of species are small, so we can learn how to do even simple experiments. But the hope is that we will apply that, we will try that on communities of different complexity. Ideally, we will learn some general principles. And I believe there are general principles to be learned because the variation and the heritability are the key issues of this process. And once we learn some general principles, then we can apply this to unknown. I mean, that's what my passion is really at. is to apply to unknown communities, right? Communities where you see a function, but you don't actually know what's going on. It's complex. And then apply this selection to improve the function. And then by comparing the evolved community with ancestral community to actually learn mechanisms about how the function arises.
So we're out of time. There are two more questions, Wenying. So if you could answer them directly in the chat and in the Q&A, both from Will and another question from Sylvia. But unfortunately, I think we're out of time. So we're going to move to the next speaker and have you comment. Is that correct, Janine? We have 5 minutes for questions?
Yeah, 5 minutes for questions after each talk.
Got it. Okay, so I think we're going to quickly move to the next speaker, which is Alison Sugrue from Boston University.
All right, so I am really excited about how groups of cells work together and particularly compute together. And one of the organisms I really focus on does this in a transition from unicellularity to multicellularity. And I think it's a really exciting question, and I want to sort of break down for you how I think about it, what are the grand challenges, or what are things we can learn and potentially use about our approach to apply this to other systems to learn general principles of how groups of cells can work and compute together. So the organism I work with and I find very exciting is the cellular slime mold. So this is an organism that is unicellular when it has food and lives in the dirt, but when it runs out of food, it transitions to a multicellular aggregate. And so what you're seeing here on the left side of the screen is how the cells are thinking about signals they're receiving from other cells as they go through a developmental process to form this structure you see here on the right side of the screen. They're using these signals that they are receiving and interpreting in order to come together through aggregation. And as they aggregate, they're differentiating into two different cell types. Spore cell type that will be able to robustly withstand starvation for a long period of time, and a stalk cell type that will be able, holds up the other cells so they're more easily be able to be carried to an area with more food. All right, and what underpins this process, in fact, I think this is a central problem, not just for cells that come together for multicellularity, but even for cells that stay together, that are part of a multicellular organism that doesn't have to come together in order to create this organism itself, is coordination, right? Cells have to generate signals, send them, receive them from their neighbors, decide what they mean, and encode that information internally to decide what to do with them, to do something within themselves. Right? This process of coordination is central to multicellularity. And I want to call out a few sort of inherent questions about it. Like, what are these challenges they face, right? If you've got two cells, right, they have some critical pathways within them that they use to interpret information. And they also need to decide what is important to send between them, right? If you think about life as a cell, right, what is it important that cells need to know about other cells? What level of detail is important? Right, and so these are some of the questions that I think that pop up when we think about how do cells work together and how do they process information as part of these groups. And one of the things that I think is really powerful for thinking about how this could possibly happen isn't just asking, what are the signals we see or what are the phenomena we see, right? But taking an iterative theory experiment approach to have a conceptual and mechanistic understanding of what are ways that cells can do this, not just in one system, but in many systems. Right? So again, I want to, broadly define what I mean here by theory, right? I mean anything from the conceptual models that sort of give us a framework for understanding general observations, all the way down to mathematical models that describe phenomena or biochemical, be these general phenomena or even biochemical mechanisms that we can use in tandem with observations of any level of detail in order to contextualize, predict future experiments, and then abstract our understanding across systems. So what I want to talk about today is a case study in the social amoeba, the cellular slime mold. And I want to sort of generally ask the question, what do single cells need to do to coordinate together? Like, what's important for multicellular function? And by what I mean, what they need to do, I mean, what are key behaviors? And then key features inside single cells, these computational units for information processing that allow them to work together as a group. All right, so dictyostelium coordinates through synchronized oscillations, right? We see these in space as waves. And something I think is actually really neat is that this is not unique to cellular slime mold, right? We see these kinds of oscillations in yeast. We even see them within ourselves and our heart cells, right? Where traditionally in good function, you see target waves, and sometimes if they're interrupted, you see spiral waves. But to sort of get down to the most basic way that a cell is seeing this, when a cell sees this spatial thing pass over, the spatial wave pass over it, it looks like an oscillation in time. And so people have been able to see these waves through various visualization mechanisms for decades and been able to measure the biochemical components that make these waves. And so they've been building models that are designed to recapitulate population oscillations through biochemical measurements for a very long time. as people were able to take better measurements of dynamical phenomena through being able to observe how single cells process information between them, they're even able to build further models. You might be thinking, oh my gosh, this is a 10-minute talk and you just had the nerve to throw up all these different components. Don't worry about anything in the details of any of these models. All I want you to take away, right, is that there's a bunch of them. There's some bottom-up and top-down approaches. And what's key about them is they all make different assumptions about how single cells could be processing information, and in particular, the network features that allow this to happen. All right, and because they make different assumptions about how single cells behave, to build up this idea that population oscillations are important and to be able to recapitulate those oscillations, that's going to have different implications for what are ways cells can process information to work together, and what are ways cells do process information to work together. All right, so multicellular behaviors in this system are more complex than just oscillations. This is a very high density video I'm showing you where you see waves over and over and over in a short time period. But if I had a low density population, they wouldn't be able to sustain those waves across a whole population. All right, and so experimental measurements have shown that you can sort of tune this coupling between cells and how well they're able to oscillate together by tuning things like cell density or a flow rate of media over the cells. And so there's a transition between these two that the models need to reproduce. And in particular, they've shown that in this regime where you have high coupling through density, but a low flow rate, in a biochemical medium, cells aren't able to synchronize, right? The external medium we think can't keep up with the internal dynamics of different cells, and so they desynchronize. And so this is a neat feature that maybe isn't important for multicellular behaviors in general, but is potentially important for all systems that need to coordinate through an external biochemical medium like this. So I like to think about, why is this behavior important? What is it allowing us to do? And where could it pop up? So would it be important in other systems or just here? So again, these multicellular behaviors seem very important because of what we're thinking about for multicellular systems, but it actually turns out, right, that what's critical for building these up are, again, how do single cells function, right? You can't have these kinds of behaviors unless the single cells are able to do a sort of information processing that allows them to emerge, right? So what's important at the single cell level for recapitulating these kinds of behaviors in a model are first, adaptive spiking in response to a stimulus. So each of these single cells, if you stimulate it, will produce its own spike and return to baseline, and that's really critical. The second thing that's really critical for models to reproduce, to be able to reproduce observed population phenomena, is having single cells act as fold change detectors. So what I mean by this is that single cells, regardless of what concentrations you put in, will respond with the same response. Something about what do single cells need to work together, and what I mean by that is, like, if you think about how single cells are behaving in these kinds of systems where you're trying to come together, individual cells may need to coordinate over many different orders of magnitude and group size, a few cells or many, many, many cells. All right, and so this is a critical feature of how a single cell needs to be able to sense information about other cells in the environment around it in order to be able to work together in this kind of system. So it turns out multiple models can reproduce these critical population behaviors, all right, that facilitate a transition to multicellularity through biochemical communication. All right, they actually take different approaches to the features that are parts of their network, or they're totally different approaches, all right, but both of them are sufficient. And so they both suggest routes that organisms could use to coordinate through a biochemical medium. Only one model is able to reproduce slime mold and its signaling behavior specifically. So it turns out a behavior that isn't important, that we see at single cells, but isn't important for populations, is bifurcation to oscillations at high levels of external signal in the medium. This has nothing to do with population behaviors, and so I didn't dive into detail. But what I think is important here is I think that this is important for thinking about an approach that you can take to think about how can multicellular systems work together. Something I think is also very cool here is that this model is also used to describe excitable neural and cardiac phenomena. So I told you that this is a model that's great for describing biochemically coupled systems. Yet surprisingly, we see this general phenomena across the board in systems that actually also stay together. So despite it being a potential good mechanism for coming together and coordinating coming together, we actually also see this for staying together systems. And I want to sort of elaborate on why this is important and what we can take away from this. So how could we possibly integrate experiment and theory to identify how multicellular life coordinates? How could this be an approach that we can take and you took the other systems to learn something in? Right? You can imagine that we observe emergent multicellular phenomena, particularly computation. All right, we can go ahead and use theory to build multiple models that describe this behavior. What are many, many different ways we could build it up using different models of single cells? Right? As we begin to get data on how single cells can behave and observe how they compute, we were able to narrow down what are possible ways you can connect these single cell behaviors to multicellular phenomena. All right, and theory really gives us this link here where we ask, what are, again, many possible ways. We don't have to build these systems, we only have to build them computationally, and we're able to then link potential single cell mechanisms to multicellular mechanisms. All right, and finally, as we're able to, again, take these single cell measurements and population measurements, we're able to define a model that best describes a biological example, and that tells us how that system does work. But we also, through this process, learn how systems could work. Right, and so that's important because it suggests best practices across the world for how you can coordinate multicellularity that we should be looking for somewhere else, and not just how this one particular system does work. It also gives us potential routes toward creating synthetic multicellular life. Right? These are all mechanisms that we could be using that can create the multicellular phenomena we care about. And maybe they're ones we aren't seeing in nature, but that doesn't mean they can't work. Something I find very exciting about these models and the results from slime mold is that both models, D and E, for example, only D represents slime mold, but model E is also a potential route to coordinating multicellular behaviors. We might see it elsewhere, but models D and E are actually only a few of the possible models that allow you to robustly obtain fold change detection, this thing that could be very important for microbes in general. And so we should either expect to see it somewhere else, or maybe we should be taking advantage of it or other features that it has in order to be able to create multicellular life synthetically. So I told you that tools are very important, and this was one of the calls for the workshop, is thinking about what could we design and how can 4D-CP help there. And so I want to call out this idea that we need to understand what's important for cellular coordination. What was enabling in slime mold is tool development for understanding how cells are thinking. But a challenge in general is that we don't really understand what cells are sensing and what's going on in the extracellular space and how they're communicating. So we don't have a great sense of how much detail they're putting into the environment sometimes. We also don't have a great sense of what they're able to sense from other cells in some systems. I used to think about this as how cells were thinking and talking, but I really realized a better analogy is how cells are thinking and smelling. Right? Cells are exquisite. They have many different receptors. And these receptors are primed to sense very specific things at very specific concentrations or very specific levels of force or other kinds of signals. And so what we really need in order to be able to build up these kinds of models and move toward a more mechanistic understanding from a conceptual one are toolkits that are able to measure and perturb across a full range of concentration scales for a number of different molecules of interest. And you might be thinking, well, we can't do this for every molecule in the whole known universe. On one hand, I would also suggest, and it appears in many systems, there are master regulators and actuators that are used over and over. We see these like calcium, cyclic AMP molecules that pop up over and over and over and get to be repurposed. And so I hope somebody talks about the potential role for how evolution acts upon these at some point. But I think in 4D-CP, this is somewhere where the program could really focus on choosing model organisms that are going to have a synergistic toolkit development. And that would be really natural for comparing for similar coordination problems. This is somewhere it could make the greatest impact, allowing a number of questions to be tackled with new tool development. And then again, building conceptual theory to bridge our understanding. So with that, I'd really like to thank the team that did the work I talked about today and take questions.
Fantastic talk, Allison. We have time for a couple of questions. I think maybe I'll read out the question from Xinjiang. And I think the question is, could you possibly also see similar oscillations that you showed in calcium signaling or any other signaling pathways?
I'm sorry, could you repeat the question? I broke up for a second.
The same oscillations that you showed for CAMP, could you also see them for calcium signaling or any other signaling pathways? Are there multi-layer communication networks?
Yeah, in this system, we do see coordinated calcium oscillations that appear to be coordinated. There also appear to be coordinated force waves. So it isn't clear which one is causing which one and how this is wrapped up. This is partly due to the challenge in multiplexing tools and separating these signals out temporarily. But yes, so that's true in this system. And then of course, there are also many systems where you see coordinated oscillations. And again, the challenge of what's causal versus not is unclear.
And then related to a big picture thing that you highlighted that we will definitely bring up in the discussions, I think you really highlighted the role of not a model, but number of models, because models give you a way to think not just in this system, but broadly thinking about other sets of systems. I think this will be a common theme that we will talk a little more about. But in that framework, I'm curious, with the waves propagating, is the motility of a single cell of the same order of magnitude as the speed of the wave? And so is a single cell seeing a periodic signal or actually seeing much more of a turbulent signal because it has its spatial heterogeneity and motility? And if you could crank that motility parameter, what would happen to the spatial information pattern that you might be listening to?
That's a great way of putting the question. I haven't heard it framed that way before. So initially, they're actually not super physically responsive. And so it's a good assumption. And something I think is also important to call out here is they actually have memory. So they don't move backward with the back of the wave. They actually sense this initial change, and then they're not responsive for a time. And so that's key for forward motility. So there's a lot of beautiful work on that. Over time, they do get faster. When they get fastest, it appears to be at a time when biochemical communication, these waves are less important and being used more to synchronize gene expression. They actually start to use physical coupling. Some really lovely work just came out that showed you can break all cyclic AMP external signaling. And as long as you turn on the appropriate gene expression pathways, if they're touching, they don't actually need to use these waves externally at all. So I think by the time we do get into that regime where you have this order of magnitude transition, that's not what they're using to physically move with each other.
And I think we have run out of time again. Allison, there are a couple of questions in the Q&A, so if you could handle those, we will jump to the next speaker. And next we have Fred from University of Groningen. And Fred also happens to be I think probably the only card-carrying philosopher in our community. So I'm very excited about your talk, Fred.
Yeah, can you see my screen?
Yes.
Okay. Actually, I'm not even a card-carrying philosopher. I only work there. But still, yes, I am a philosopher, and this talk will be quite different, I suppose, from all the others, although not too different, because many people talk about concepts, and that is what this talk will be about. Does this work? No, this works. But I'm a philosopher, and what I want to talk about mostly is about conceptual work. How do we interpret the world that we see? And love, we also can say. Do we see a rabbit or a duck? Depending on whether we see a rabbit or a duck, we focus on very different things. The back head of the duck is not significant, but the nose of the rabbit is, in that way. If we use words like cheater, cooperator, and things like that, they influence what we see. So that is general. What I want to focus on is what is questions in general, because philosophers love those questions. And in this case, I want to focus on the question, what is a nervous system? But in line with the multicellularity evolution question, I will focus on the evolutionary perspective. And this is guiding into a different direction from what we usually think of when we talk about nervous systems. We don't talk about nervous systems at all. We use a different concept, the brain. The brain is the source of our intelligence, the intelligence of all animals, basically, and we use it in a very offhand way, and we think we understand what it means, namely the thing that helps us or makes us sense the environment and provide enabling us to act. And this is something that we also think of single-celled organisms. So if we focus on paramecia or other protists, and we focus on planarians or hydra or other simple animals with nervous systems, we don't think too much of that too easily. Somehow the nervous systems are there to make the animals intelligent. Just here is one illustration coming from Herbert Spencer Jennings, who already wrote in 1906, and this has not been sort of retracted since, is that when it comes to behavioral capacities, sensing, and many other features, there is actually hardly any difference between the single-celled organisms that you investigate and the basal animals like planarians, hydra, and so on. They are not more complex. So suppose that the brain or nervous systems are not there to make us intelligent, or these organisms intelligent. What are they there for? And one thing of the things that I want to sort of bring into the open is that having the same functionality but at different scales makes sometimes, and in this case definitely, a huge difference. Building blocks on top of one another, or putting blocks on top of one another, is trivial in the one case but very hard and requiring a totally different organization to accomplish it in the other way. And it might very well be that when it comes to nervous systems, they don't make, or didn't make initially, these animals more intelligent at all. They just make them do possible tasks that they could already done, or other organisms could already do at a single scale, but now at a multicellular scale. So this is a very different view on what nervous systems did, or at least initially. So from the single cell to the multicellulars, there is an evolutionary path connecting the two. We are here, so it must be so. But it does not need to be a very direct path, and the connection might be very much that of a very complex labyrinth that has an entrance and an exit. We don't know where that is. That was already mentioned earlier in an earlier talk, or at least we must know what these entrance and exit points are. But it also is a very convoluted road that, and this is sort of more general remark compared to in relation to the other talks, that we cannot think of as something that can be easily generalizable. It might be that there has been this very precise sequence of steps that led from one to the other, at least in the animal case, because there's only one lineage in which animals came up with nervous systems. Okay, so what happened in between? One way of thinking of this is in terms of lineage explanation, a term drawn from the Australian philosopher Brett Calcutt. And basically what he stressed was that we need sequences of complex organisms that functioned. So there must be a continuity of different organizations, different multicellular organizations in this case, or initially single-celled maybe, depending where you want to start, that became complex and different over time. Here, with the eye case, it's the functionality of this specific feature is simple because it is all in generating a stable or a good image. But when it comes to organizing animal multicellularity and animal bodily organization, the functionality of its components might have been very different at certain stages. If you think of how we evolved our lungs from swim bladders of fishes, a swim bladder function is very different, or is a different function than what it came to when animals got out of the water. So this is something that we don't know, or we don't know what the functions were in between, but this is important when it comes to making sense of this transition. So back to nervous systems and their functions, because there will be many. A possible question at this point is what was the crucial function of early nervous systems that later enabled the evolution of animals as we now know them? And the crucial function I put in there because there will be many different functions that have, because these nervous systems are extremely useful. But what might have been a crucial function? That's the focus I want to zoom in on here. And then it makes sense to consider two options. On the one hand, the input, what I call the input-output view on neural organization, which is the standard view. You've got some sensors like eyes, you've got effectors that move the animal about, and the most simple nervous system that you can think of is just a direct connection that sets the effector in motion. This is why we tend to think of brains in general, an input-output function, basically often similar to a computer. The thing that the interpretation, the function that I want to set in opposition, or at least in addition to the input-output view, is what you might call an internal coordination view on neural organization. Basically, what you focus on here is that nervous systems may have been essential in getting muscle contraction, getting contractile motion organized in the first place. Muscle contraction is something that involves the whole body. It must change shape, and you have contractions. The contractions are necessary at specific places at specific time in a dynamic way. Getting this organized, and think back about sort of building Stonehenge, is a task in itself, you might say. And this comes then into the form of a coordination proposal. Starting out with individuals, they become reorganized in ways that may be nice or less nice. But what is going on is that somehow, in the animal case, early nervous systems must have helped to tie cells together into mechanically unified bodies, capable of moving as a single dynamic unit. So instead of free and happy moving cells, we became tied together, maybe still happy, that's not the point, but we became tied together and had to move and coordinate in a coherent way to act as a single individual. So the key function in this case is patterning contraction activity across the body or the muscle surface as a whole. It's a different function. It must have been essential, you might say. It's unclear in what sense it's related to other possible functions of nervous system, but it was sort of important. So it's plausibly, I'm sort of sidetracked now, yeah, plausibly an evolutionary stepping stone that eventually enabled large-scale and forceful action at the multicellular level. It enabled large-scale ways of sensing at the multicellular levels. We just saw the question about what do cells sense, and they might smell and they might feel the tensions and the mechanical forces of other cells. But we, at least as very complex multicellular animals, organisms, can do much more and can sort of sense a very different environment, like looking at this screen at this very moment. And this is all done by connecting mechanical, chemical, and electrical forms of signaling at cellular scales and connect them to happenings at macroscopic scales. So somehow this connection must be made to make us even possible in the first place. So what is a nervous system? The question I started out with. Well, as I already implied, it is a very complex question and there are numerous answers, not one of them necessarily the only good one. But being a coordination device is one answer that is too often overlooked because it is so self-evident. But if you think of how to move this large doll and how many people it involves, it is definitely very complex. So thanks for listening.
Thanks so much, Fred. That was fantastic. We'll open to questions. And while we are waiting, maybe I'll start off with one of the threads that I love in your work, Fred, is it makes us think quite a lot about scaling in multicellularity, because we are now exploring not just the transition point of the origin, but we're talking about systems that could go from hundreds of cells to trillions of cells. And so, I mean, I think in terms of even different kinds of nervous systems, do you want to, either from your prior thoughts or just real time, say a few words about context of nerve nets versus more organized nervous systems from an internal coordination perspective in the evolutionary tree of different kinds of approaches to this long-range coordination? I'm very curious about, as animal bodies get larger and larger, this kind of a coordination resource at very long distance becomes more and more relevant. And is there another transition point in nervous systems, much more from distributed nerve nets to a much more centralized organization which works with nerve nets?
It's a very good question, difficult. basically, I would say don't focus on the getting extremely large in the first place, because this doesn't happen in any way very easily. There must be sort of a sort of architecture organization present to allow this to happen in the first place. So when you so focus on NerveNet first and sort of going to simple brains. Basically, what you can say here is that we think of, I showed you the human brain, and the reason for that is that it forces us into thinking in a specific way, which I think is giving us the impression that it's not actually super complex or super not understood at all in the first place. We have a computer metaphor. We are sort of totally familiar with entities that think and make decisions and do stuff. That's not an issue. That's not a problem. But when you sort of get ourselves out of the picture as entities that have these capacities and interpret other entities in this way, then you have no clue how this might be possible. And what I like about this coordination view is that you can always bring in a computer, but computers don't build themselves, they were built by humans. So how do we come up with this transition? And what I now tend to think of is that what happens in brain is actually not very different from nerve nets. Basically, what you see, patterned activity across a surface, or maybe a volume, but the surface is easier to think about. And you have a limited number of muscles that happen or contract and extend in certain patterns. This is a way in which an extended surface can be sort of make a connection with happenings at a molecular cellular scale, I think. This is very vague, I know. But what you need is sort of ways of thinking about making this connection, how you get from the very small to the very comparatively millimeter, centimeter scale, quite large. And how this can be done. With a nerve net, it becomes possible to think of making connections between cellular, molecular level happenings and slightly larger ones. If this is in place, then actually, if you've got a nerve net that sort of is capable of doing things, it doesn't matter that much how it is configured, because being a net is not essential then. It's about connections, about entities, units in the nerve net, and you might condense parts of the nerve nets, which is sort of self-evident because it's cheaper energetically, and it might condense into sort of, well, small brain.
So,
which is actually
Fred, we're out of time. Okay. I wanted Eric. Eric, did you have a quick question? Or we can bring that to the discussion session as well.
Yeah, very quickly. Do you perceive that the phenomenon of regeneration might give a clue as to how assemblages of cells coordinate, i.e., there's data from Elliot Tanaka which suggests neurons are needed to regenerate the amphibian limb. And there's also data from, I think, Redian, who suggests that in planaria, very long-range muscle fibers are sort of a mesoscopic organizing center in planaria when they regenerate. So these are all sort of long-range coordination mechanisms, and maybe for the discussion you could reflect on whether that is also along the same pathway to the neurons that you suggested.
Yeah, very short. From this view, or many people think are sort of going into this direction, that the difference between neurons and other cells in signaling and making things happen is not that large. They're actually specialized, but not so specialized. So basically, that would be the short answer.
And then Eric, let's bring that up in the discussion, because what you raise is really the multifunctional nature of these sets of systems itself. Since we're out of time, I want to jump to Eric's talk. So Eric, if you could share your screen. So now we have Eric Siga from Rockefeller University.
The screen is visible?
It's visible, yes, perfect.
Very good. So I construed this session, which had theory in its title, Once Upon a Time, and I will address the question of what happens when you cross biology with physics/mathematics. among the offspring that are produced in that union are the legitimate ones, where I think the physics is overt, such as structural biology, single molecule mechanics, things that are the purview of the biophysical society. I would prefer, however, to concentrate on the illegitimate offspring, where the physics is more covert, and that will be the topic of my talk in the context of development, doing some theory and also experimental systems. You all know from much data, an example being this book of Bateson, that development is canalized, i.e. if you have a moth where the leg turns into a wing, you get a anatomically correct wing. You don't get a linear combination of leg and wing, and similarly for the thumb to fingers. Another example of canalization is the famous Spemann experiment, where you take a bit of the dorsal lip on a frog, transpose it to the ventral side, and get two very symmetrically formed head and torsos. Again, you get a reasonable looking head and body. You don't get some linear combination of dorsal and ventral. So the phenomena of canalization invites one to do phenomenology. That is to say, one can inquire as the gene network that turns a leg into a wing, but you do not have to inquire separately or in addition how to make a wing, right? So it's a modular system. And that, so if one goes to the opposite extreme, bottom up and reductionism, you would tend to write kinetic equations for all the genes in your network. And these equations are completely indecipherable to anyone, mathematician or physicist or biologist. The same is true, you may not know, that when you write down the equations of Newtonian mechanics for the solar system and include the interactions between the planets, the long-term behavior of that system is also not understood. So the way mathematicians deal with these types of problems, which are very common, is to not tell you how a particular solution evolves in time, but to simply classify the types of behaviors and then get a discrete list of the qualitative types of behaviors. In the case of the systems that would be relevant to development, there is actually some sophisticated mathematics that goes from the flow description, which is arrows in a space of genes, to topography. That is to say, one can, with some technical caveats, show that the Waddington landscape metaphor is in fact a theorem. In addition, the mathematics shows you what are the operations that will trivialize this topography, and in that sense allow you to enumerate a couple of categories of cell fate transitions in geometric terms. To fix very crudely the sort of ideas here, on the left I show you a flow field, and all that means is a visual representation of the dynamics. There are points such as this source from which arrows emanate. There are points here such as a sink where arrows converge, and there then must be in between other points, saddles, where some arrows come in, some arrows go out. And of course, you could imagine that this set of arrows could be mirrored by a topography, a topographic map, where the same features are shown and cells just follow gravity. And that construction is indeed quite general. Any such flow field can be shown to give rise to a topography. Now, the interest of that, among several, is, for example, in using these schemes to fit fairly high dimensional and complex data in relatively simple terms. So one instance of this is where we have, looking at a stem cell system, looking at stem cell transitions beginning from the epiblast to various specialized cell types. One can pigeonhole, the data allows you to pigeonhole the cells into discrete types. You can apply morphogens, and then you could simply categorize the cells as a function of time and morphogen, apply morphogens in the morphogen history. But when you do that, what happens is that you see the topography changes as you add these morphogens. Cells flow from one fixed point to another fixed point, and eventually settle somewhere at the bottom. But these transitions then give you a quantitative graphical representation of how these cells respond to those signals and give it to you in a form that's easily interpretable and sort of intuitive, even though the real cells involve, these signaling pathways involve tens or hundreds of genes. To go on to my second little topic, which is to say, you could all imagine how we could take cells in a dish, apply morphogens, signaling pathways, and push them to various fates. We also want to investigate how do tissues self-organize, in particular epithelial tissues. And I would like to advertise very, very superficially how embryonic stem cells can be used to build synthetic embryos, and in that way understand these signaling pathways and self-organization from the ground up, i.e. if you could build it, you could understand it, sort of following the Feynman-esque mantra. We have done this beginning 8 or so years ago by the very simple stratagem of simply spatially confining and organizing the stem cells to these micropatterns. Shown here, the bigger patterns have on the order of 2,000 cells. And what you could see is that these patterns, when you expose the cells to uniform morphogen, BMP4, which we know from the embryo is what kicks off the signaling, kicks off the development, you could see after two days fairly reproducible patterns with various cell markers that are indicative of the embryonic germ layers. There's a great deal of work in this area now, and let me just say very, very succinctly that the surprising fact from these assays is that once you control the geometry and density just a little bit, the topology of the 2D patterns mimic those of the 3D embryo around the time of gastrulation. That is to say, in the case of the mouse embryo, where we know most quantitatively what the morphogens are involved, 10 years before our work, mouse embryologists were quite willing to reconceptualize the signaling in the embryo as this disk system, where what is next to what is preserved as you flatten the cup to a disk. And that is indeed what we found with these human embryonic stem cells. Now, there's, again, a great deal of work in this area, but I would claim that these synthetic systems are the ideal compromise between fidelity and accessibility. That is to say, in these systems, you have BMP, Wnt, Miller, FGF pathways all working together, all working in space, self-organizing, all with their secreted inhibitors, all of which are moving. So all of that is going on, and you can't make it any simpler and still be vaguely faithful to what goes on in the embryo. But nevertheless, these pathways do self-organize in this crazy synthetic geometry in a way that is as faithful as possible to the embryo, given the topography. There is also interesting cell biology of how signals are internalized and secreted in these epithelial layers, which have been sorted out in these synthetic systems, and some of which has been checked in real embryos. And finally, of course, we do 3D models as well as 2. So to conclude this very broad brush summary, I remind you that genetics over the past many decades gave us the parts list for embryonic development, but has left us with a very myopic view of how to assemble those parts into an embryo. Stem cell systems, I believe, are a unique way of seeing how those pathways interact in a somewhat natural environment and in a way that's very easy to quantify and manipulate and measure. The phenomena of canalization permits modeling by degrees or levels. And the mathematics to which I alluded to gives you a very short list of, so to speak, dynamical mechanisms that lead to cell fate transitions, which you could then fit to data in a fairly succinct, compact way. The biggest conclusion, and I guess the one most relevant to the rest of the meeting, is that the most useful data is not at what comes out of the screen, which is great for illuminating the parts, but in fact should be dynamic, i.e. you don't want to measure the endpoint, but you want to measure the dynamic process when the cell is jiggling among those various saddle points, deciding to go, so to speak, right or left, and eventually emerging into one discrete fate. So this requires a model to interpret and design the experiments, in contrast to what my immunology friends at Rockefeller tell me. Your experiment cannot be simply, are the mice dead or alive, but in fact, what is the dynamics that led to their demise or their behavior. And therefore, one, for purpose of development, one definitely needs dynamic measurements, movies, and their interpretation has to be meshed with models to interpret what is going on, and thus fit and extend to other situations. So with that, I am done, probably even in 10 minutes.
That was perfect, Eric. You were right on time. We have lots of time for questions. I'll start with a question from Buzzbomb. Eric, does the success of the 2D in vitro models of development imply that patterning is really a 2D problem? That is, embryogenesis is origami?
Okay, that's a very good question. And as you would imagine, is rather more context dependent than I let on. In that PNS paper I briefly cited, there are examples of proper spatial patterning. The 2D thing works well in the situation where you have discrete endpoints, fixed points, so to speak. And if you're looking at the transition between fixed points of which everything is attracted, what sits between points where everything is attracted are points where n-1 things are attracted and one is unstable. So in that sense, it is low dimensional. However, there are circumstances that you could easily imagine, such as bristle patterning with long-range Nash delta, where there are many more unstable directions because it's a sparse pattern induced by pair competition and pairwise inhibition. So it depends. But the function of these models for dimensional reduction is very strong and an important part of their utility and their comprehensibility.
So while we are waiting for another question, maybe one question for you, Eric. I think the mathematical framework that you described, especially taking these attractor flow networks and also a map on the potential contours, could you give an example in the context of data? Because I'm assuming in the framework of dynamical systems, you can find a certain class of data. These potential class of models have been utilized in the past. Is this mathematically rigorous map between the two?
Yes. Okay,
could you just say a word about that?
Okay, that's an important question, again, dealt with in that PNS paper recently. It is not, people have used potentials before, and people such as René Thom, of mathematical distinction, have used potentials to look at local bifurcations. What is new is actually using work from many decades ago in pure mathematics from Smale, which was designed to prove the Poincare conjecture. And in that context, he showed that any flow, with some minor technical, any flow is equivalent to a potential and a metric, any flow. So that's why I said the Waddington metaphor is indeed a theorem. And that is rather potent statement when you go to fit data. You're always allowed to put a potential, and the potential also allows you to look at global bifurcations, not just the local bifurcations of Thom. And those are important in the data fitting, even in that example I showed you. I didn't obviously remark on it, but that data with the stem cells involved a global bifurcation, which is in the list.
No, that's fantastic. Zev, I see your hand.
Yeah, I have a question, Eric. That was really great, and I love the way you put a lot of things together there for us. And I just have one question about the final point that you raised, and it's really just sort of a technical question, which is if the strategy that we need to take to really understand the shape of these potentials during development is to make these dynamical measurements, do you envision that being done through microscopy? And what ideas do you have to, if so, what ideas do you have to think about how to deal with the just enormous increase in scale of data?
Right. Okay. So
that we're going to need to.
Right. So that's a very good question. I think in some cases for these instances of spatial patterning where you have reasonable markers for cell type A or B, simply video microscopy with those markers will get you very far because it really focuses on the dynamics. And of course, those types of problems can be made very sensitive because you could start the model with the actual crazy initial configuration of those particular cells and then look for what the final pattern is with your markers. The data I showed you from the stem cells, which was single cell data, was simply high dimensional FACS data, but it was taken at fairly tightly spaced time points with a good repertoire of time-dependent signals applied to flesh out the phase space of what all those morphogens could do at various times. I mean, it's a big parameter space when you think of morphogen levels, morphogen times, and the various cell types involved. And of course, if we endeavor, as we advertise in all our grants, to say, do regenerative medicine and actually engineer this or that organ, we do have to make predictive models and then fit modular data to these models and then glue the models together. I mean, the parameter space to make some sort of organ from stem cell to even fetal organ is too vast to contemplate, but you have to fit pieces of it together and you have to have a quantitative model. So a bit of both data, but I'm not a great big fan of drop-seq and RNA-seq, these very high dimensional things. I've learned a lot from simply high dimensional FACS, which you can do it many, many times and it's fairly quantitative.
Yeah. So we'll take one last question from Otkar. I know, Ginny, we have a break. I don't know how long the break is going to be. So this discussion will just continue in the break itself. If you could just say how long the break is, and then I'll let Otkar ask the question.
Yeah, let's just take 5 minutes and then we'll resume the next talks. So everyone can take a 5-minute break, but you can go ahead.
Yeah, we'll just utilize this time. It's too exciting to not let people chat. So Otkar, go ahead.
Hi, Eric. So quick question about the stem cell differentiation process. If you have very well-defined markers, I understand actually that you can reduce the problem, the dimensionality of the problem, and just look at the stem cell marker. But in general, when you're studying this, you need to look at a very high dimensional space, and we all face this same problem. It's like, how do you reduce the dimensionality of these spaces in a meaningful way? And this problem actually has been discussed now recently a lot, actually, in the scientific community. How do you do it properly, actually, to reduce the dimensionality? So what are your thoughts about that?
Okay, very good, thank you. It's a very good question, and it's certainly system data contingent. So in the case of the stem cell system I showed you very briefly done with Briscoe, there he has proper high dimensional RNA-seq data, which shows that there are relatively well-defined clusters of cells done in the full genome expression space. And these various intermediate cell types that from development are indeed real from the point of view of single cell expression data. But that has to be checked for the system in question. In another instance, say hematopoiesis, which is always drawn as a nice linear differentiating bifurcating tree, of course that data is largely, I think, from cell markers, not from full single cell data. So it's I think, to my mind, open question, to what extent are there discrete states in any of these differentiation hierarchies? And of course, what that I showed you, what we call, quote, mesoderm, is of course going to ramify, we're just doing germ layer specification. So of course, beyond mesoderm is all the types of mesoderm. So obviously we restricted our focus to markers that distinguish mesoderm, endoderm, ectoderm. But I think that, one has to check in perhaps a high dimensional space whether there are these distinct types. And if there are, you could then model those decisions among those distinct types. The other thing that the geometric models also give you is they give you a functional form in which to fit what you think are the irrelevant directions. So, and that's useful because you're fitting, all these things are noisy, right? How much noise is noise and how much is real? Well, you have to fit data. So you get from these geometric things, you get functional forms that allow you to fit the irrelevant directions, or what you think to be irrelevant directions. Of course, on top of that, there's all this issue of how if you homogenize someone's brain and you look at cell types, that's of course a tough problem of classification. But I think if you do it through a developmental pathway, it's somewhat easier. But as you say, it certainly is a fair question as to what extent are there ever discrete types until you get to the very end of development.
And do you think that if we do dimensional reduction, because many people like to do the dimensional reduction and make this 2D plot, the bifurcation plots actually they were showing, do you think that there are maybe cases in which that dimensional reduction cannot be done? And actually, even if you were to reduce to the minimal, to the central manifold, let's say actually of the system, actually, that central manifold would be larger than 2 dimensions.
Okay, fine.
You could not reduce further than that.
Okay, fine. So we give in that PNAS paper a very simple example where you're making a sparse array of bristles on the back of it's real data, it's back of a fly. And in that case, you manifestly have a saddle point with 8 unstable directions. Manifestly, because there's that much flexibility in how, with repulsion between the individuals, repression between individual cells, you could pattern this 2D thing with respecting the inhibition. So that, but again, putting it in those terms allows you to parameterize the data in as condensed a way as possible, but certainly there are many more than 2 unstable directions. It depends on the problem.
Thank you.
But the geometry makes it as simple as possible and no simpler.
So we have a minute left, and I think, JLS, you had a comment. In the meantime, Corina, if you could just test your slides, and in parallel, JLS, you could ask your question. And either Eric can answer now, or I'll pick that up in the discussion.
So Eric, that was really great presentation. I really like the fact that these micro patterns are reproducing your circular symmetric gastrulation. What I'm curious about, you've been describing this system in terms of adhesive and repulsive interactions between cells. To what extent do you think that, again, mechanics, physical forces, or nutrient availability in these systems, as these cells get more crowded, as they're being pushed, they can't expand out because they're restricted in different ways. To what extent do you think that really is setting up the whole system?
Okay, that requires a very long response, but just very briefly, certainly in these mechanically, spatially, they're confined because of ECM. There is certainly, as defined by availability of beta-catenin and stress fibers, mechanical forces around the boundary, sort of barrel staves. These systems are still very deficient in sorting out morphogenesis. An interesting example recently is from a guy at UCS in Santa Barbara who has produced a neural tube and neural tube closure, and he is getting his hands more on the mechanics, and he is 3D. So that is a weakness of these models, but people are filling that gap. In terms of the sort of, we could also grow these epithelia on porous filters, and we could see very long-term stability, but manifestly these things are popping cells out continuously.
I see.
So lots is going on, and it's just a very good system to sort things out in a simple way, but still somewhat realistic.
And then let's take this, Eric, in the discussion a little bit as well. I want to make sure we give Corina the time.
Clearly.
So maybe, Janine, you could let me know if we can start the
go for it. Let's do the last couple of talks.
Okay, so I think we have the pleasure of hosting Corina Tarnita from Princeton.
Okay, great. Well, my brain is on fire from all these super exciting discussions. It's been such a wonderful meeting all day. So my work focuses primarily on the origin of biological construction, and I try to take a comparative approach, not just within multicellularity, but across other kinds of hierarchical transitions. So also the transition to eusociality, and typically the kinds of questions that we ask when we look at these origins of construction is, first of all, we want to understand what makes construction robust. Why does it stick around once it happens? But then, also questions of how does complexity arise and how does diversity arise? And I'll leave these in question marks because, it's a bit in the eye of the beholder who is more complex and in what way, but in principle, something more interesting that wouldn't have been there at the origin. So typically, until about a decade ago, the frameworks that I learned, the very broad conceptual frameworks that I learned to deal with these kinds of questions, kind of started with, well, we have a fundamental problem of we have a group of cells, and when we have a group of cells that aren't yet used to playing nice with each other, we have to worry about cooperation and control of cheating. We have to worry about how to keep these cells together and instead of them kind of disintegrating. So the robustness, if they can do that for some time, then some magic happens, and that magic is some sort of integration. And if they are then together, and perhaps at this stage we can call them a unit of selection, going back to what Wenning was introducing, then eventually some sort of complexity might arise, for instance, differentiation or division of labor. And because, at least at a very broad conceptual level, this magical step has been pretty hard to tackle, often the question has been boiled down to how do we get things to cooperate? How do we suppress cheating in this very kind of broad and abstract realm? And so there's been a lot of work done around mechanisms for the evolution of cooperation, and certainly a lot of work done on how things get assorted with, like, for example, kin selection or assortment with individuals who are your relatives. And there's been then a lot of interest in trying to understand, the importance or postulating the importance of things like the unicellular bottleneck that happens in clonal development but doesn't happen in aggregative development. Now, this was until recently, and there's been some really interesting work that's been done there, but I would say that what has happened over the last decade has really changed the game, and theory is still kind of lagging behind a little bit in terms of actually putting together the new conceptual framework that integrates work such as the one that Iñaki was talking about this morning. And so I would say that over the last decade, as a theoretician, the things that have gotten me extremely excited have been the novel results of comparative genomics. And I would group these into, first of all, the accumulating evidence for the important role of the ancestor. It's not a boring, spherical thing that we put in a group and then we wonder how it can cooperate. It's a really genetically interesting thing that exists in a really possibly interesting ecology and can do a lot of things and might have to do those kind of temporally sequential, but once you put it in a group, interesting things may happen. Many more interesting emergent things can happen. And there's, of course, the possibility for co-option. And so here, the work of Iñaki and of Nicole King has really kind of got me thinking. The other one has been disproving, again, in these very simple model organisms and simple phylogenomic studies, or elegant phylogenomic studies, disproving the widely assumed idea of a certain intuitive order from less to more complex. And here I'm thinking about the work of Pauline Schaap, for instance, or Matt Heron, and showing that things such as germ-soma differentiation, which for the longest time we thought you need to take a really long time and play nice for a really long time before you can do something as sophisticated as germ-soma differentiation. It turns out that if you look at slime molds, for instance, germ-soma differentiation has been lost many times to actually lead to acetylsterids, for instance, where each cell contributes a little bit of cellulose to form the multicellular fruiting body. So starting from these super interesting advances of comparative genomics, I think there's really scope now for rethinking sort of from a bottom-up approach that highlights the ancestor just a little bit more and allows it, instead of being this spherical, uninteresting object, to possibly, endow it a little bit more with its own, let's say, gene regulatory network, allowing it to explore its own environment. And therefore also, once we do that, put in there the environment and think about what kind of ecological context would this ancestor have existed in. And think a little bit about this ancestral stage before allowing for the evolution of, or for the origination of a group. And when we do that, that also brings about the idea that, of course, we then have to start thinking about how the group came up, right? And this is actually reviving an old idea of John Bonner, which he introduced in, already in 1965, that it's really not a good idea to think about multicellularity in the absence of the ancestral life cycle. The first group will have originated within the life cycle of the ancestor and will just be one stage within that life cycle. And therefore, group formation and propagation and the mechanisms by which those occur. And I have here two, just using the broad terms of staying together and coming together for what many of you might refer to as clonal development and aggregative development, only because I want to apply it broadly also for social behaviors. So staying together is the same idea of you start from one unit and then that divides, and that's the new complex. Coming together is you have independent units and they come together and they form something. Now, just a very simple example. So we've been toying with these ideas and how to put together such a framework for a few years now. Just to give you an idea of the power of just putting these two things together, a more interesting ancestor in a more interesting ecology, and then putting the group as a proper stage in the life cycle of the ancestor. So group dynamics is explored there on the left, then giving it a very kind of simple, for the purposes of this particular slide, but I'll come back to it, a very simple benefit, ecological benefit, and that's just that a group of a certain size will be able to escape predation, a group of smaller size than that will not. Then I can search a parameter space that tells me, where does the benefit of multicellularity kick in? And the other axis is a physical property of the ancestor, how sticky it is depending on, again, just a physiological constraint of the ancestor. Just by putting in these very, very simple, so we have an ancestor with a gene regulatory network who lives in a variable environment, and at some point, one of the genes it expresses in that environment makes it sticky for some small period of time. We can recover 6 different types of life cycles. So a diversity of life cycles, not all of which are, trivial. In fact, some of which are at a very simple level of comparison, the counterparts of extant life cycles. So in red, we recover, for instance, or in this pink, whatever color this is, we recover, the cycle of the metazoa. You're always multicellular, but every once in a while you split a propagule. But we also recover something like the slime molds in brown or yellow. And so diversity can arise actually pretty early, and this kind of bottom-up approach can show us that you can get interesting things very quickly if you allow the ancestor and the ecological context to be more interesting. And this is what this is saying. But in general, one of the things that we've noticed theoretically is that the evolution of this hierarchical organization requires that the benefits of group living actually arise at very small group sizes. And I would say that maybe one way to think about that is that you could think of it in terms of predation. So you get the benefits of predation at fairly small group sizes, but you could think of it more broadly and then ask, how? So how, in more interesting ways, rather than just escaping predation, would a group have a very high benefit from being, or would cells have a very high benefit from being in a group? And here's, some of the comparative work that I was mentioning with other origins. Here's a very elegant and simple experiment that we did in collaboration with Daniel Cronauer's lab at Rockefeller, where we saw that if you put ants, and these are extremely simple ants, there's no queen, they're parthenogenic, they all lay eggs and they all take care of those eggs. So they exist in the wild in very, very small group sizes. What they actually manage to do when you put them together in groups of different sizes, we see that if you have just one individual, it mostly fails. It's not even able to take care of one more offspring. You put 2 individuals, it starts to do a little bit better. But as you increase the group size at around 8 to 10 individuals, you start to see really quite a high benefit. And eventually that benefit actually plateaus at 12 and 16 individuals. So what gives this really sharp, this boost from being in a very small group to being in a very small group, but just a little bit bigger? what we found in these particular ants is the ability to spontaneously self-organize. And what they do is actually rudimentary division of labor. I call it rudimentary, and I want to emphasize that because when we think about the origins, it's often better to rethink what we call division of labor. If we were to measure it with entropy and other measures that we have for like really sophisticated groups, we might miss the kind of really interesting but very rudimentary division of labor that these groups are able to do. And I'm happy to talk about that in the questions. So bottom-up approach is it's in general allowing us to see that there are many things that can happen in small groups. You can have synergistic interactions depending on the latent potential of the ancestor or the genetic, interesting genetic complexity or whatever is in the ancestor. You might get really interesting things come up for free. I'm going to skip a simple example on robustness here, or my vignette on robustness, but I would just want to emphasize the fact that a bottom-up approach that kind of is informed by these novel recent advances allows us to get at question 3 from Inyaki's slides. It allows us to explore the possible and not just the actual, not just the realized, because we kind of allow it to explore a much bigger space and not guide it that much. It might get us to reframe old questions. For example, yes, cheating might still be a big thing, but what cheating actually is, once you put it in this different, richer context, will have to be rethought a little bit and might lead to much more interesting ways of thinking about it. Sorry, I don't know what happened there. And it might also show that some conceptually hard questions may actually have been easier than we appreciated in the past. For example, some degree of spatialization might come for free at the origin, but that at the same time forces us to rethink what was actually hard. No one is arguing that things were completely easy at the origin of these various major transitions, but it forces us to rethink how to place these questions to find out different angles to think about what was hard. And I just wanted to put here what I think are the kind of key elements, because I'm already over time, but I might leave this up as I take questions. I would say that before we get to the fourth part, which is how selection acts on this new unit and what new emergent properties arise, new work has shown that there's scope for a lot of interesting stuff to happen. But unfortunately, there haven't been many frameworks or models for that matter. It's been kind of serendipitous, taking information from here and from there and putting together models that show us really interesting new angles to think about it. But it hasn't been done in a framework that conceptually puts it all together. What is interesting to me? What is interesting to Iñaki? What was complicated from, or hard from Eric's point of view or from Lucy's point of view, right? And how do we bring all of these perspectives together in a now more coherent bottom-up framework? And then take sub-models of that we can start inspecting specific questions in like smaller bite-sized versions of putting these together. And I'll leave this here with apologies that I've taken a bit long, but yeah.
No, Corinne, we have time for maybe 2 quick questions. I think there is a question in Q&A from Rensky Romans. Thanks for the talk. In the model of evolution of life cycle, what caused the group sizes to typically remain so small? Does it have something to do with the time scale of environmental fluctuations, or is it really a cost-benefit balance?
Oh yeah, that's a good question. So in principle groups, there's a cost here that I took out so that I don't because in the interest of time. But if you have big groups that are too big and haven't figured out a way to transport nutrients, to figure out how to do other more sophisticated things, we assume that there is a cost to be paid to being into a too big of a group, because cells on the inside, for instance, might not have access to resources. So there's a you shouldn't be getting too big. That is constrained by this cost. Good question.
And then there are 2 other quick questions. One from Israt. Really interesting talk. I wanted to ask if you've looked into reversal of complexity from diverse opportunities available at origins freely. Things going a little bit backward.
Yeah, we have not yet, but of course that's a really interesting question. I mean, you can see already a little bit of that in these life cycles, because you might argue, well, this life cycle where groups can grow and grow and grow and they never fragment, they never fall apart, except every once in a while they put out propagules, might have the opportunity to discover other things that are more interesting. But at the same time, there's also this life cycle that just kind of decides to dissociate completely every time that the environment changes from one environment to the other, right? This is more like a slime mold kind of life cycle. And I think, you know, you could argue about whether you could see these life cycles as a loss. You know, one of them is kind of allowing for more things or staying together for longer periods of time. Maybe more things might happen. The other one kind of falls apart so much. But we haven't explicitly explored the question of loss because, yeah, no, I think we should.
And then Iñaki has a comment, which I'll bring up in the discussion because I want to make sure we give Jacob enough time. So last but not the least is Jacob Muser from EMBL. Jacob, take it away.
Hello, yes, let me share my screen. Can you see my presentation?
Yes.
Oh, that's the wrong slide. Let's go to the beginning slide. There we go. All right. Hello, everyone. Yes, thank you very much to the organizers for inviting me. My name is Jake Musser, and I'm a research scientist in Detlev Arendt's group at EMBL in Heidelberg. Much of our interest in the Arendt Lab is on understanding the origin and diversification of animal cell types. And I think in the context of understanding multicellularity, particularly in animals, this is a really interesting topic. The challenge we really have, though, in understanding multicellularity and its origin in animals is that most animals have thousands of cell types that arise through quite complex developmental processes. And so we've been developing sponges as a comparative model for really two reasons. One, for understanding where animal cell types and cellular systems come from via the comparison between sponges and really anything else in animals. And also really as a model for sort of what's the minimal requirement of what you need to have a fully functioning animal. And I think this bears directly on questions that 4D cell physiology is going to be asking. Sort of what do you sort of need as pieces that have to be able to integrate and cooperate to make an animal? So sponges only have about 10 types of cells. It's been very difficult in the past to make comparisons between sponges and other organisms because their body plans are so different. So in this is a cutaway, SEM cutaway of the sponge that we work with. It's a freshwater sponge. Sponges are essentially filter feeding animals. So they have a complex canal system that they pull water in through. And you can see this sort of overlying epithelial tent that has holes in it. So water gets drawn in through this into sort of subdermal lacunae and then pulled through this canal system before eventually exiting through the osculum. And there's sort of three main tissues that comprise the sponge that you find these different cell types in. The first are the pinacocytes. These are epithelial cells that form this tent as well as the lining of the canal system. These are thought to be contractile. We have mesenchymal cells that include the main totipotent stem cells called archaeocytes in sponges, as well as a variety of other kinds of mobile cells like amoebocytes. And then lastly, and importantly, we have choanocytes, which form these chambers within the sponge. I'm highlighting a few here. These are actually embedded in the canal system, and they're important in both digestion and driving water flow through the system. So water is flowing through the canal into this choanocyte chamber. The cell bodies are around the periphery, and they have both a microvilli collar and flagella, which is helping drive the water flow through the system. So this eventually goes out through this sort of apopylar pore into the excurrent canals. So we've been using approaches like whole body single cell RNA-seq, where we essentially take apart the organism, which you can do in about a minute. So it's very quick with sponges to dissociate them. And then we capture them in microfluidic droplets and profile their transcriptomes. So I'm showing you here data from about 10,000 individual cells. This is one of these 2D plots that I heard people mention in one of the recent talks. What you're essentially trying to depict here is some sort of gene expression relationship that is some sort of similarity-based information between cells. But what I can sort of broadly summarize here is that we've been able to define about 18 distinct expression programs. These include the archaeocyte stem cells, as well as transitional states that go out to all of these different sort of differentiated cell types on the periphery. Interestingly, this sort of differentiation cascade is quite simple in these organisms, at least from what we can see in our data. So it really seems like you sort of start as an archaeocyte, and then you shut archaeocyte genes down to turn on the genes of your differentiated program. There's not really a lot of distinct, in fact, there's really only one sort of distinct intermediate progenitor, and that is these choanoblasts, which are proliferative and are at the beginning of the choanocyte chamber formation. The other thing we've been able to do is we only have 18 cell types, so it's very possible to sort of see the spatial relationships of all of these different cells within the organism. So we've used approaches like SNFISH and HCR to track down all the different cell types. And I'm just highlighting you here an example where you can see the excurrent canal with all of these different choanocyte chambers. In magenta is a gene expressed by choanocytes. Oops, sorry. And in yellow is a gene expressed by these apophylar cells. This is a closely related cell type of the choanocytes that forms the excurrent pore. The other way we've looked at our data, though, is to look at how these sort of differentiation programs are organized. What do they share in common with each other? In our bodies, if we think about how cell type expression programs are organized, we're not all sort of equally dissimilar, right? We see sort of families of cell types. We see different types of muscles or different types of photoreceptors or different types of neurons. We wanted to know, do we see something similar in sponges? And in fact, we've been able to show with phylogenetic methods that in fact sponge, sort of the differentiated expression programs are organized in an internested hierarchical manner. And there's really 4 sort of broad classes or families of cell types that sponges have. And I'm highlighting here these 4 that really represent, as well as sort of the genes that sort of characterize that group broadly. So you have a group that includes the stem cells, the archaeocytes, and related cell types in sponges. You have choanocytes and their digestive machinery, an amoeboid neuroid immune family, as well as the pinacocytes, which I mentioned are contractile, indeed are broadly expressing a lot of muscle and contractile genes. One of the things we wanted to do with this, though, is then to take this information and to ask, okay, so there's sort of some functional similarities here between sponges and what we see in other organisms, and actually try to decode whether there is, in fact, whether we can actually say, for instance, that pinacocytes are more closely related to, say, muscles genetically than they are to other types of cells. And so for this, in collaboration with a group at Stanford, Bo Wang's group, as well as a student in his lab, Alec Tereshansky, we've developed a method for aligning single cell data sets across really distantly related species. This takes into account complex gene relationships that often, I think, hinder our ability to make comparative statements across phyla. But in this case, we've also been able to show that within phyla, where the homology relationships of cell types are kind of undisputed, it does a very good job of sort of recovering these relationships. And we've applied this to the sort of existing or growing number of whole body single cell data sets that have been published. This includes a number of vertebrates, sort of early embryogenesis up into organogenesis, as well as several flatworms, the Cnidarian hydra, and our sponge data. And in fact, with this, we've been able to really show that genetically you find best alignment between, in this case, sponge contractile pinacocytes with all of the different muscle types in these different data sets, as well as the contractile epithelial muscle cells that you find in cnidarians. And this is being driven by actually thousands of genes. So we can also pull out the genes that are sort of driving this. And we can see that there's actually quite a few genes, including both on the regulatory side, so transcription factors, several of which are sort of core hub proteins in the regulatory complexes driving muscle cell type differentiation, but as well as that, the sort of all the components of the contractile apparatus. So beyond just sort of actin and myosin, right, things that are also involved in, for instance, junctional proteins that are helping to bind these together. We've also seen concordance if we look across stem cells, particularly in invertebrates, strong connections, also driven by many genes. So this includes sponge archaeocytes, as well as eye cells or interstitial stem cells in hydra, and neoblast in planarians and schistosomes. And interestingly, from the perspective of sponges, which don't seem to have a nervous system, we actually do get alignment of some of our cells with neurons. And these are actually our digestive cells, the sponge choanocytes. And this is really actually being driven by the presence of postsynaptic scaffold machinery, as well as a number of different receptors and ion channels that are highly enriched throughout the whole choanocyte family and close relatives. And that's really noticeable that the postsynaptic genes are very localized to this group. In contrast, the presynaptic genes, particularly the secretory apparatus, really seems to be found much more broadly. So we find it in subtypes within the endymocytes. Several of these are the contractile family, Several of these are probably sensory and secretory sort of subtypes, as well as even within choanocytes, we see secretory machinery. And in particular, a gene, a cell type that really stood out to us was this cell that we've called a neuroid cell. This is actually a historical name that was given to the cell. We think we've sort of rediscovered it. That is really upregulating both immune genes because it's part of this sort of immune family, but also many genes involved in the presynapse and in secretion. And so it was particularly striking to us when we looked at the spatial relationship of these cells, and we found that the digestive choanocytes, which are expressing sort of the postsynaptic receptor machinery, have a close physical relationship with these immune secretory cells. So these immune secretory cells are actually living inside the chamber. This is actually external to the organism. You almost usually have one or two per chamber, and you almost always find these sort of just in the choanocyte chambers. And so we've used a combination of X-ray sort of volume imaging with focused ion beam SEM to localize and look at specific chambers. And this is a segmented and rendered video I'm just going to show you here. This is a choanocyte chamber. You're looking through the X-current pore. We're going to rotate around the chamber. Now, most of the choanocytes here are grayed out. These are the digestive cells around the edge. But we've highlighted a few in color that are part of this interaction between the secretory immune cell and the digestive choanocyte. So as we're going to zoom in here, there's actually two of these secretory cells, a magenta and red one. And they're actually both reaching out projections that reach into and seem to actually enwrap or entangle with the microvilli of particular choanocytes. Now, I don't have a slide for this, but these cells we've been able to show are packed with secretory vesicles. We do not see a synaptic junction as far as we can tell. Nevertheless, we do see, these secretory vesicles, and they actually, you can even see here this cilia coming out. They form pockets around the cilia as well. So almost encasing the cilia. So we think there's a role here for communicating about either the microbial environment or potentially regulating feeding within these animals or water flow as well. And so, this sort of, I think, provides a new picture potentially of neuronal origins, because one, it shows that things like the presynaptic genes are really broad. But two, it says that the place where they do come together is not necessarily muscle, but actually seems to be around digestive chambers and around maybe the microbial interface. And so, it's interesting to note this and what this implies for nervous system evolution. I think we're still developing, but nonetheless, I think this has been an exciting finding. I'll just leave with sort of one last conceptual thought as we go to questions, which is that, when we look at these comparisons really across these distant groups, we don't see sort of one-to-one cell type homology. We don't see an alignment of sort of these single cell types, but rather really groups, whole families of cell types seem to sort of broadly be homologous with each other. And I think this is consistent with a model in which you have sort of a limited number of cell types ancestrally in animals. We propose perhaps something like a sensory contractile, a neurodigestive, amoeboid immune, and a stem cell, perhaps more than that as well. This may even go back prior to animals when you had cell cycle or life cycle transitions. But nevertheless, each of these lineages has then diversified it further within animals and in different lineages. And as a result, you don't have the sort of one-to-one homology across these sort of phylum distances. So with that, I'll just thank, there's many people who are involved in this research. We've had a little bit of a sponge team in the Arendt lab. And so it's been a pleasure being here and happy to take your questions.
Fantastic job, Jacob. We have a couple of minutes for questions. While the questions, Eric, go ahead. Eric, you can unmute.
Yes, sorry, guy, I have to unmute. I'm sluggish. Is there any suggestion in these sponge cells that there is electric depolarization as a signaling mechanism? Do you see specialized ion channels or pumps or things of that ilk that might, or you do calcium imaging?
So they do, and so we haven't done calcium imaging yet, though that's coming. They don't have shaker channels, so I'm not sure they can do a classic action potential. Nevertheless, they do have differential expression of both calcium and some other ion channels as well, both potassium and chloride. We don't know the function of these yet, but yes, absolutely, I'm sure control of ions is So we can manipulate calcium and we can induce, so sponges do these whole body contractions. So there is a role for calcium in regulating this contractile tissue. But yeah, beyond that, we don't know.
Thank you.
And I think there is a question from Iñaki. I'll just let him ask that question. Iñaki, do you want to unmute and just ask?
Sorry?
I know you have a question in the chat. I just wondered if you want to ask it directly.
Yeah. Okay. Yeah, Jacob, thanks a lot. Maybe I had already asked this to you before, but anyway, sorry if that's the case. So do we know the function of these presynaptic and postsynaptic genes in sponges? Because, you know, they may be, I mean, whether they do the same as in bilaterians, because I'm wondering if they may be doing something different here. And, you know, so the interpretation may be a little bit different. Thanks.
Absolutely. I think that's a great question, right? I think this gets at sort of how novelty in cell type and cell function arises, right? And clearly the synapse is a highly integrated unit, and that those pieces have not been fully integrated, even if they are doing something similar in sponges. What I would say is this is an area that I think, well, let's get to where I think 4D cell physiology and where the future needs to go. You know, we've talked a lot about gene expression, and a lot of our comparative biology has been based on gene expression. One of the things we're really moving into is interaction proteomics. So using approaches where you can globally profile protein interactions to start to look at that. And in an organism like sponges, you can then intersect that with single cell data, and you can start to, you know, you have a simple enough organism in terms of cell diversity that I think you can start to piece down to cell type specific functional networks and understand, you know, how and where those exist within the organism. So that is the future. Yeah, absolutely.
Thanks.
And I see Israt has a question, but I just want to ask Janine first, how much time do we have left for discussion? Because I'll let Jacob, you answer that question in the chat. While Janine, can you tell us how much time we have left for
I would
discussion?
say 20 minutes for the discussion.
Okay, so since that's a short time, Jacob, if you don't mind answering that question in the chat. And I just want to open up the floor to firstly, all the panelists. I mean, I think the framework idea was very important to us when we were thinking about this group of people to share and talk. You heard everything from what is a question all the way to multiple overlapping approaches to be thinking about these broad sets of questions. And I think it would be very valuable if, firstly, the group of panelists or certain sets of folks who have burning questions that connect the pieces together, you could raise your hand and I'll kind of walk through that. I'll kind of kickstart this because multiple people said this in the context of what I find incredibly beautiful in this problem is the multi-scale, which is fundamental to understanding biology, shows up in a very modular manner. I think all the way from units of selection, all the way to many modular models that Allison had talked about. And then finally,
go to another meeting.
I think, okay, I think there was some interference, but so maybe for the panelists first who covered these sets of frameworks, if people might be interested in talking about this hierarchy and this notion of challenge with scale, and especially if there are certain frameworks and theoretical approaches that allow us to handle this challenge of scale. For example, I was quite struck by Corinna's, your last example in eusociality, that the experiments didn't recapitulate the kind of scale that I see that ants cooperate in. Maybe we're not measuring things, or are there fundamental limitations when we talk about that these sets of approaches will be able to understand communities that are beyond millions of cells working together? So just anybody who wants to jump in on thinking about scale and sets of challenges that both mathematics and these sets of frameworks still have to resolve.
But sorry, Manu, I'll ask you to just, can you clarify what was it that it was inconsistent with? Like, what was your expectation?
My expectations was that eusociality would include another orders of magnitude of complexity in that system. And I saw a curve that was plateauing between 10, 15 organisms. So maybe you could start from the largest scale of literally trillions of ants working together. What are the commonalities? What are we missing in our tools to truly understand that large number of ensembles? And maybe we'll take steps back.
No, that's such an interesting question, right? Because for me, the more surprising thing was that 6 ants can have the same kind of, benefit that 12 ants and 18 ants will have and 30 ants will have, because that means that even 6 ants that were genetically identical, I didn't have time to mention that, were able to somehow act like a little complex system. And they did that by doing this rudimentary division of labor that Iñaki asked about, and I will just take a moment to say what it was. Well, when you have 4 ants or 6 ants, we actually started to see like a proper boost in fitness at 6 ants. When you have 6 ants, they can't say, I'm going to be a nurse and I'm going to be a soldier and I'm going to be a whatever, right? Everyone has to do a little bit of everything, right? So they'll be on a continuum. They'll all be doing a bit of everything that needs to get done. What will start to become conserved over time as you get to a certain group size, but not below that group size, will be the ranking. So how much of a nurse I am today versus how much of a forager you are, right? So I may do a little bit less nursing tomorrow and you might do a little bit more nursing, but I will still be more nurse-like tomorrow if I'm more nurse-like today, right? So it's a relative ranking on this continuum spectrum of work that becomes conserved, right? And this is what Manu is pointing out now, is exactly the kind of question that then, on the one hand, it's really great news. A small group of genetically identical ants can do something really cool, can spontaneously self-organize, right? But then how big does that group need to get before you start to see other cool stuff happening, right? And then you start to get really into other like very cool questions of, let's say, genetic heterogeneity or architecture, right? So like interactions within smaller groups can cement certain roles and make them more efficient in that particular role. So there's a lot of super interesting stuff that you can ask after that. But to me, the main thing was that, wow, 6 ants can really do division of labor, when 4 ants can't do that, and they can do as much as like 16 ants can. That's cool. Mm-hmm. Because what boosts the fitness of the group by that division, by this very rudimentary division of labor, is that it increases homeostasis, which made me think about questions of, earlier talks in the morning about like inside-outside boundary and the question of homeostasis and the question of what can happen, as you start to form like a group that is of a certain size.
Mm-hmm. I know, Wenying, you have your hand up. Maybe if you would want to comment both on the perspective of scale and also where do you find limitations in our current mathematical frameworks for these multi-scale problems?
Actually, I have a question for Corina. So
yeah, go ahead.
For the ant problem, right? So did you actually try to have many more ants than the ones you tested? Like maybe thousands, maybe tens of thousands. Do you see anything cool emerging from like many ants? And then the second question, in the natural environment, what is the group size you observe? Or maybe there's a range of group size.
There is a range of group sizes, but they are not the kinds of ants that you would see with like 1 million ants. They will be more, you will find them with 100 ants or 200 ants, right? In the lab, there's a limitation to how many you can I think you can keep on a petri dish. Daniel would speak better to this, but I think the biggest group size that they managed to maintain was like 32 or something like that. Interestingly, I don't think we saw more. Yeah, I think it might actually even have been 16. It's a pretty complicated setup with,
Okay, so then,
but this is.
Now we'll shift the question a little bit, right? So if you have a group of individuals, it's actually impossible for them to be identical. I would say not being identical is more a natural state. And have you thought about theoretical conditions under which, if you have heterogeneous phenotypes, right, that you will have a better group function or worse group function, right? Under what the conditions the group function, whatever way you define it, would be better if the genotypes or phenotypes are heterogeneous versus homogeneous, right? Because we have, we also have this bed hedging. So there are this genetically identical cells under the same abiotic environment would show different phenotypes, right? But to deal with uncertain conditions. So what are the, so I'm just curious about conditions under which, right, this heterogeneous phenotypes always were more helpful or always less, more fit or less fit in whatever the group function you define.
I don't know if that was a question still for me, but I will just say that yes, it's very much something we think about. And this was actually a follow-up paper with Daniel's group because they can control things so well in this ant system, because they're parthenogenic, we were able to control, they were able to control different kinds of heterogeneity, genetic heterogeneity, so make groups with genetically heterogeneous, make groups with different sizes, different numbers of ovaries and other things that distinguish these ants. So we could different ages, so we could properly explore heterogeneity. And actually the paper just came out this year. There is definitely scope for more interesting, in everything that we've seen so far with these ants, there's scope for more interesting stuff to happen. And I am trained, or I had initially learned about heterogeneity as being much more of a challenge, to like integration but actually I think there are ways to look at it to see a lot of like benefit, especially when it comes to spontaneous self-organization. If you have things that are slightly different and can take slightly different roles, you might very quickly get the kind of division of labor that might, you might only need 4 ants to do that and stuff like that. So yeah, but I can send you that paper. I don't want to take up the whole,
Yeah, I think we have 10 minutes left. So I want to edge towards the direction of, if sets of people find what you found missing in what was presented in the frameworks. And I know Lucy's hand is up and Amy's hand is up. But I would be very curious for people to push back a little bit and think about, what do you want theoreticians to work on? What do you want for their tools to actually be useful directly to your system? So this is your opportunity to really grill them all collectively in terms of what is missing. Anybody wants to take at it? But of course, if you have quick sets of questions, you can also shoot them too. But I'm curious if somebody has a burning desire in, is this enough in this context of framework? And of course, panelists can jump in as well, because you're all thinking about what's next. So I'm curious if anybody wants to take this invitation.
Sorry, Manuel, are you asking about the theoretical tools or theoretical models of interaction with theorists, or what do you mean?
Yeah, theoretical tools and frameworks, for example. I mean, I think framework is a much broader perspective here. I love the fact what Eric described as a fundamental link between one dynamical approach to this landscape approach. It suddenly opens up a new path. And I'm just curious whether people find that immediately useful. How many of these types of approaches are there? Because, questions are so dramatically different. But again, this is literally also about what's missing. And of course, go ahead, Eric, and then Jacob.
One item which I think is sorely missing that we all encounter is readily reusable image processing software. Amelia had a very good professional, Gene Myers, on the staff for a while, who was very much interested in, he began life as a genome sequence assembler, but thought also a lot about the image processing. I don't know what came of that, but there certainly is a dearth of general purpose tools. Everybody is doing the same damn cell segmentation thing over and over and over again. And there are parts that we use from whatever, MATLAB, whatever, or Python, but one is lacking the sort of general robust tools that we have for genome assembly in the image processing, and we waste a lot of time doing that.
Mm-hmm. Yeah, no, I think that's a point very well taken. And there's lots of groups at least focusing on it at this point, with Napari and a couple of tools coming online. Jacob, do you want to add to that, go in a different direction? I'm just curious of what do you find missing?
Well, conceptually, I wanted to go in a little bit different direction. I wanted to bring back actually to one of the points you made originally, which was the role of environment in all of this. we have a tendency to bring these organisms into our lab and play with them and try to remove effects as we can of this external environment. But, clearly in the transition from, into multicellularity, I think there's a huge role for this. And I think some evidence for this comes from some of the work of Nicole King's lab, where they showed that you can give antibiotics, right, to these choanoflagellates, and you can affect sort of cell transitions, their cell identity. And you go into then multicellular organisms, and you seem to have, I think, lost some of that plasticity. I think, and in fact, it's actually really a change in plasticity that's accompanying this transition into multicellularity. You have to be able to sustain a gene expression program, right, or an identity, despite all this other crazy stuff that's going on around you, right? And not to say that there isn't plasticity in cell types or multicellular organisms, of course there is, but I think this sort of idea of thinking about it as changes in plasticity, or going back to even an older concept, the reaction norm, changes in the reaction norm as the way that you are accessing multicellularity or different cell types is a framework that I think we need to go back and think a little bit about. And I'm always interested in ways where we can find, avenues to explore this, and would love to hear what others in the panel and listeners think about this.
Yeah. Does anybody want to directly comment on incorporating a variable environmental world directly in models to Jacob's question? And then I'll pass to Lucy. Go ahead, Karina.
I just wanted to say that I think, I'm in an ecology and evolutionary biology department, so I certainly spend a lot of time thinking about ecology. I think all of these models, we're trying to put a little bit more into it and a little bit more into it. What has been really interesting to me was to start to read books by earth scientists and geoscientists and start to think about the paleoecology. Paleoecology, so like how much can you really think about predation as a major force versus, let's say, synchronous migration, right? Or other kinds of things that might be happening. And so that I would love to spend more time thinking about how to even properly, once we acknowledge that thinking about a variable environment is super important, and I think I think that should be acknowledged, then exactly what that is and what is it variable in, I think is non-trivial.
Yeah. Allison, did you want to follow up just on that itself?
Yeah, I wanted to follow up on the environmental idea a little bit. And
go ahead, and then I'm going to pass to Lucy. Yeah.
Absolutely. Just very quickly, I think the other thing that pops up to me is also this idea of spatial environment, particularly with things that are, coming together. This is inherent. Karina brought this up with like the dish and, ants on a 2D substrate in a petri dish. The same exists with microbial communities that we've heard about today, or, this is a problem with slime mold. We're looking at models, but we're also looking at behaviors that we have brought into a simplistic lab environment, both, biochemically, but also physically, how it is spatially structured. And so we're potentially getting out these mechanisms that actually aren't the things that we're selected for in any way. I'm putting this out there as a both, theoretical challenge, adding all of these layers of spatial scale as a big challenge, but also, this is an experimental challenge. How do we even see these behaviors in a natural context? I know this is something all of us struggle with resolving, but I think it exists a challenge on both parts to understand how is this acted upon and how is this evolved is something we need to also work towards solving.
I'll pass to Lucy and then to Zev. Go ahead, Lucy.
Yeah, actually, maybe a pair of comments. I mean, with this idea of bringing in, trying to model the extracellular environment, Allison had such a fantastic point earlier about how there's a dearth of tools for real-time monitoring of extracellular signals. And, like if you can't measure it, it makes it hard to monitor. I mean, when we think about trying to measure nutrient availability, we don't know what the local concentrations of the nutrients are. And so something where, imaging tools for that would be great. My original point was coming back to cell plasticity. And Karina made me think about if we, these different roles that the ants have, if we think about that as a very rough analogy to cell fates, and Jacob making the point that as organisms became more complex, we've lost a lot of plasticity in terminal cell fate decisions. I'm curious, Karina, whether in your modeling or in your experiments, you mentioned being able to create ants with like extra ovaries, for example, if you're can lose some degrees of flexibility those ants have in the roles that they're able to take on, and what effect this has in terms of optimality of a certain group size.
Yeah, that's such a, I want to first clarify that I can't make anything like basically other than write equations, but the people who do make these things. I think one thing to be clear, we can't, they can't make ants with different numbers of ovaries. It's just that there is natural variation within the colonies that they keep in the lab, and then you can put together in an experimental setup, because these ants can actually play with each other, even if they have different genotypes or they have different sizes or whatever. You can put together ants that were some that have more ovaries, some that have fewer ovaries, just because you know that that's how you select them, and then you make a new group and they still, right? So I can't say, I mean, it's a really interesting question, but I would have to ask the people who, but yeah, that's how you have a source of, because as Wenning put it, that source of variation exists. Even in these really clonal ants, you still find they will have different ages. Obviously, they will have slightly different sizes. One has one more ovary than the other. But I certainly take that work very much and kind of make these same parallels to cell fate, yeah, and what phenotypes or right, the plasticity of the ancestor. When I think about multicellularity, I just gave that as an example because I think it's nice to show that you can really scale. And going back to Manu's point, it is very hierarchical. You really do find the same questions with all these different organisms.
We only have a minute. I see, Wenning, you have your hand up.
Yes, I want to say something to theorists.
Yes.
Okay, so it seems like for us, like the very basic evolutionary questions. For example, if we have multiple tubes of E. coli growing, and then in each tube, E. coli is also evolving, and so the fitness, there will be a mean and a variance of intra-tube, mean and variance. So there will be mean and variance, and then you can ask, how does the inter-tube variance change over time? Seems to me it's a very, very basic question. And then there are theoretical papers, I mean, theoretical physicists working on that, and they publish those papers, right? But they were written in a way such that even experimental physicists could not understand, right? And it's just a really, really hard barrier. And then we were forced to ask various people to help us. And it turns out, oh, you have to go to this, complex analysis. You have to go to this, right? And then I don't know, like somehow we need to think of a way to lower these barrier of communications between different branches of, between math, physics, and biology. And I think that's critical.
Yeah, I mean, I think that's just such a beautiful way to structure it. And since we're out of time, I want to put Fred on the spot in this perspective of what Wenning asked, because I think you had a very simple question. You said, what is the question to begin with? And I think just framing that question so well for what a nervous system is really allowed. So Fred, after hearing many of these talks in multicellularity, does a simple class of questions emerge in your mind when you kind of look back at a big picture? And I know I'm putting you on the spot and giving you sort of a chance to close this session also in a more philosophical tone. How would you synthesize, as Iñaki had put, let's work on the most important questions? And I kind of find that too personal. And I feel like, I don't know if we will agree. And it worries me. So maybe Fred, do you want to chime in on this challenge? And maybe it's a much more philosophical challenge.
No, of course I don't want to, but you put me on the spot. So now actually I feel an alien in this circle, in this field, because what I'm used to, the kind of discussions, it's much more vague and higher level and so on. And so I just don't understand some of the problems you're talking about. Say, why can you not upgrade an ant colony to much larger sizes? I think, well, this is sort of the fusion rates about, say, you've got this big ball of ants, which is 10 meters across. How would they breathe inside? And so you need a plumbing system and all kinds of conditions. I think the most interesting questions cannot be studied at present because they're just sort of, we don't have the tools, like the visualization mechanism, tools and such. they're very much similar to in the 19th when you couldn't study animal behavior because you just couldn't film and analyze what they did, the rodents and so on. Things have improved there a lot. So there's a lot that can help here. But what I think about multicellularity is, I think there are several problems, which is sort of getting the initial stages sort of understood. We've seen all kinds of oscillations at very limited levels. I think that's sort of clear thing to go and expand on and help us sort of give us a clearer picture in the long run. When it comes to, say, the fungi, the plants, and the animals, there things are much different and much more intricate and complex and unknown. And yeah, I mentioned this. You might approach this more in a historical way, which is the sequence of possible steps that have led to this, to the current forms. We shouldn't take our current presence as a sort of self-evident as proof that this will be sort of likely candidates for evolving multicellularity, which I think is trying to prepare my talk for this difficult audience for me. What struck me most is actually two questions. Is the interesting question that we would want to ask in a general way is, are animals to be expected? Conway Morris once published this book, We the Expected, where evolution would sort of end up with us humans in all cases. I'm just wondering whether animals can be expected when life is already present at a single cell level at other planets. So if you rerun evolution, would animals be sort of a common feature there? I have no idea. So yeah, nobody has, of course. And the other one is sort of trying to sort of articulate the link between development, building complex body structures and organizations, and behavior itself. Behavior is, of course, much more dynamical, but there is so many similarities in how sort of single cells come together and came up to form all kinds of complex shapes, that this might have been a sort of pre-behavioral way of pre-behavior, so to say. So that's also the other question, and it's sort of super interesting, and I would have preferred to lurk only and not to be expected to have an input, but at least in this way I can lurk and learn so much from all of you. So thanks for that.
Yeah, thanks, Fred, for a fantastic way of both thinking about this. I know we are 5 minutes over time, so I'm going to pass this back to Janine. There is so much more that could be said and thought about. There is a special closed session after this, so please, I know it's been a long day with all kinds of other things pulling us in many directions, but I'm hoping that you'll all stay for the closed session. Janine, I'll pass you back for logistics. I don't know when that starts.
Yep, I'll cover it. Thanks so much, Manu. Thanks a ton to the speakers today and the discussion leaders for the really provocative talks and the conversation. Thank you also to the audience for joining us. Come back tomorrow for day 2, starting again at 11 a.m. Eastern.